Hello all, how are you? So hope you have revised your amino acid metabolism part. Okay. So guys, any doubts? Start practicing MCQs. Okay. So if you have doubts, you can ask me, right? You can tag me in the group, right? MCQs, right? So doing MCQs and all. So we were last time we were doing this heme metabolism. Okay, heme metabolism we just reached to the step 2. So what I told you, okay, the steps of heme synthesis we started. Okay, I told you that's the pathway which occurs in both mitochondria plus cytoplasm, right? Both mitochondria plus cytoplasm. Okay, and I told you that there are only few pathways in our body which occurs in both mitochondria plus cytoplasm. That is, we can remember by this mnemonic, right? PUBG. Okay, now the upcoming is 4G, so don't forget PUBG, right? 4G may be something new, but not here. Okay, so first and the last 3 steps they occur in mitochondria, rest all in the cytoplasm, right? Rest all in the cytoplasm. Fine. Okay. So I told you about the first step, glycine plus succinyl CoA. So amino acid used is glycine. Succinyl CoA is a TCA intermediate. Intermediates of Krebs cycle and their uses is one of the very important MCQ that you must know. Like succinyl CoA is one of them, which is very important. So first enzyme was ALA synthase, which was B6 dependent, which is inhibited by the glucose and the heme. That we will, use it as an application. We will see that how it can be used in the treatment of porphyrias. Okay. Then ALA synthase deficiency or mutation leads to sideroblastic anemia. Right? Sideroblasts are something like RBCs with the nucleus, and the nucleus is surrounded by the iron, the ring of iron. Okay, so it's a kind of iron deficiency anemia, microcytic, hypochromic, but with the high iron stores. Okay, with the high iron stores. Okay, guys. Okay, second step was LR dehydratase. Okay, LR dehydratase. Fine. Amount just I'll get back to you. Okay, LR dehydratase, lead poisoning. It is inhibited by the lead poisoning, right? So there will be high amount of ALA in the blood. So have you revised lead poisoning? I told you, right? Lead poisoning should be one of the topic that should be learned by heart. Very important topic. Okay, this is the biochemical portion I told you. Okay, so that is ALA levels will be high in the blood. Okay, and what we get is PBG, porphobilinogen. Now in the third step, right, in the third step, porphobilinogen deaminase, okay, which is also known as uroporphyrinogen 1 synthase. So it produces uroporphyrinogen 1, type 1. Okay, fine. Topic is heme, right? So this is the product formed. Now deficient enzyme PBG deaminase. Okay, PBG deaminase. Fine. This is the enzyme. If it is deficient, this leads to a condition that's called as acute intermittent porphyria. Okay, what do you call this? This is called as the acute intermittent porphyria. Fine, that is PBG deaminase. So guys, see the word acute. So very few cases of porphyrias which start with acute. So first of all, what is a porphyria? Porphyrias are the conditions, okay, which arises because of the defective heme metabolism, right? Okay, because during the heme metabolism or heme synthesis we are making what? Porphyrins. Okay, so if any of the enzyme is defective or deleted or deficient, so then what will happen? There will be accumulation of these products of the heme synthetic pathway, and this leads to condition known as porphyria, right? This is the one which presents with the severe symptoms. Fine, this presents with the acute symptoms. Okay, suppose a patient came to the emergency department with severe abdominal pain, right? Suppose you are posted in the emergency department and you get a patient with severe abdominal pain. So what you will think? You will rarely think of anything like acute intermittent porphyria, right? You will think of a lot of other things like acute pancreatitis or maybe like intestinal obstruction. Fine, but so this patient presents with, okay, acute abdominal pain. Fine, but guys, you will not find any pathology. Okay, so you will send the patient for, let's say, you will collect the samples like blood sample for, let's say, pancreatitis amylase. You will check the samples for, let's say, for diagnosis of MI, CK and everything you will check, but you will find nothing elevated. Okay, so acute abdominal pain with no pathology. Okay, so previously these patients were went into the unnecessary surgical exploration. Are you getting a point? Okay, no voice. Hello, hello, my voice is clear? Hello, 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 hello. Please reply, my voice is clear or not? Hello, hello, hello. Fine. Okay, fine, great. So guys, so these patients, let's say before, let's say 15 to 20 years back, such patients, when they came to the emergency department, they were found to have no pathology, right? So they were unnecessarily sent into the surgical exploration. Like, what is the problem? Like, what is the pathology? Why this patient is coming again and again with the pain and nothing is being found? So they went into unnecessary surgical, okay, unnecessary surgical exploration. So they had multiple surgical scars on abdomen. Okay, this was previously diagnostic. Okay, so even if you find a old patient in your hospital, right, with such multiple scars, okay, but now the techniques are very easy to diagnose the porphyria if nothing has been found else. And now you found that amylase is okay, so it's not acute pancreatitis. You found USG is normal, CT scan is normal, everything is normal, right? But then this is the one feature, the neuropsychiatric symptoms, and one of them is, okay, hallucination. Okay, hallucinate. Okay, so neuropsychiatric symptoms and tachycardia, which may be because of pain. So these three features, okay, 1, 2, 3. Okay, so pain with neuropsychiatric features, that can be this, but obviously you will not suspect it easily, okay, unless until the patient is known case already. So First of all, you have some more points to see here, and that is the patient's urine, okay, will have high amount of porphobilinogen, okay, porphobilinogen, and this gives wine colored urine, okay, wine colored urine sample. I'll show you one figure, okay. So wine colored urine sample, again this is the test that you can send the sample to the lab, okay. So in the lab, emergency biochemistry lab, so absorption or the urine, okay, urine absorbs light at 410 nanometers, okay, 410 nanometers, fine. So guys, this is the unique band, this is the wavelength at which the urine sample of a patient of porphyria absorbs light, especially this acute intermittent porphyria, okay. This can be any other porphyria also, but this is only in the cases of porphyrins, that I told you the structure initially, if you remember, four the heme structure is iron plus 4 porphyrin rings, okay. So the porphyrin rings absorbs light at 410 nanometers, and this is known as the Sauret band, okay. So what do you call this? You call this a Sauret band, fine. So what I mean to say is Sauret band. I'll show you one figure. See, this is, first of all, say this is a urine sample, this is the urine sample, fine. This is the wine colored urine sample of this case, okay, wine colored urine sample, and see, okay, this is the normal sample and this is the wine colored urine sample, and this is the band, okay. This is a spectrophotometric analysis, Sauret peak or Sauret band. You can see this is peaking at 410 or 20, around 410 nanometers, okay. Clear guys? So these are some unique points, features which are directly diagnostic, okay, and which are very simple and not at all costly test, okay, and gives you instant diagnosis. Clear guys? Very important to remember. So absorption of light at 410 nanometers, and this is called as Sauret band. Clear? So urine has high amount of PBG. Fine. Now the next point is, this patient has history of fasting, right? Patient has history of the fasting, okay, history of fasting. Fasting may be like more than, what is fasting, guys? Fasting means when we are not consuming food more than 12 hours, okay, that's called as fasting, okay. Ideally we take this value as a fasting as per Harrison, right? That is around 8 to 12 hours that we have not consumed anything, so take 12 hours as fasting. So why the fasting? Because fasting increase heme synthesis, okay, fasting, and this increases the heme synthesis. Now the reason behind that is very complicated, but I just want to tell you that during the fasting what happens, as you know, the glucose has to be maintained, right? So there is increase in the gene expression of gluconeogenesis, okay, gluconeogenesis enzymes, as you know, fine. So G and G, gluconeogenesis enzymes, right? Which also, so there is increase in gene expression of gluconeogenic enzymes, which also, I mean to say is, the gene, that is DNA. Okay, so this is the DNA, and on the DNA, let's say this is the genes for gluconeogenesis enzymes, and which are very close to the genes of ALA synthase. Okay, ALA synthase. See guys, normally our genes are separated. I mean to say, if one gene is expressed, the other gene is not expressed usually. The nearby genes are not affected much. But this is one of the case when the genes for gluconeogenesis, when they are expressed, the genes for ALA synthase will also express. Okay, this may not be asked to you, but this is just for your knowledge. I'm telling you, if you ask like why the fasting is increasing heme synthesis. So the reason for that is fasting increases the gene synthesis of gene expression of gluconeogenesis, right? And the ALA synthase, which is the nearest gene, right? Okay, the gene of heme synthesis enzyme, ALA synthase. Am I clear? Everyone fine? Okay. So guys, that is the reason, you can give this patient IV glucose in the treatment. Okay, so I'll come to that point later on. Okay. Okay, so this is for gluconeogenesis enzyme, like PepsiK for example, and this is ALA synthase. So they are very close to each other and sometimes expressed together. So fasting induce heme synthesis. Clear? Next point is there is history of drugs. Okay, and what drug? These are enzyme inducers. Okay, enzyme inducers, right? As you know, okay, I'll tell you 3 names that you must never forget. A, B, C. Okay, A for alcohol. Very common history. When the patient of acute intermittent porphyria came to you for first time, there are high chances that they have consumed alcohol in the last 24 hours in high quantities. Alcohol is a inducer. Which enzyme? I told you, you are done with this, CYP 450. B for barbiturates. Okay, and C for contraceptive. And when I'm saying contraceptives, it means any steroid. Okay, contraceptive. Okay, so A, B and C. Understood? A, B and C. So this is the history of drug consumption. Everyone clear? Okay. Now, what is the treatment given? Just few points I want to add. You have to give IV glucose, or you can say dextrose. Okay, someone asking me what is the calorific value of dextrose. So guys, dextrose monohydrate gives 3.4 kilocalories per gram, right? Okay, if given as a purest form. Okay, though carbohydrates are giving us 4 kilocalories per gram, but if it is given IV as a parenteral nutrition, pure dextrose gives 3.4 kilocalories per gram. Clear? Somebody was asking. This was a recent MCQ actually. So, you can refer more to your surgery department and anesthesia department, because they are dealing with the total parenteral nutrition. Okay, so it is the answer. if you want to know, 3.4 kilocalories per gram. So IV glucose and okay, and heme, okay, infusion. So guys, I told you heme is the heme is the feedback inhibitor of the ALA synthase. Okay, heme is the feedback inhibitor of ALA synthase. So guys, that's all that you must know. Very important topic, acute intermittent porphyria. So the three steps, three highly important MCQs I told you. Okay, that's heme synthesis. We are taking number 4th. Now I'm completing the pathway. Questions are now less common in exams till now. These 3 points are super important, you must remember. Okay, now there are some questions which are less commonly asked. Okay, but they have been asked. 4th. So guys, you have now hydroxymethylbilane, or I can say uroporphyrinogen type 1. So it is acted upon by the enzyme that's called as uroporphyrinogen 3 co-synthase. Okay, what do you call this? Uroporphyrinogen 3 co-synthase. So obviously what we get is known as uroporphyrinogen 3. Fine. Uroporphyrinogen type 3. So type 1 and type 3, they differ in the structure basically, and we need not to, like when we are solving MCQ, we need not to go into the depth. Okay, just you should know this basic thing. Type 1 is not found in human beings, type 3 is found in the heme human beings, right? So UP3 is formed. Okay. Then deficiency of this enzyme, uroporphyrinogen 3 co-synthase, or synthase, you can remember just. Okay, this is known as the condition that is called as congenital erythropoietic porphyria. Okay, congenital erythropoietic porphyria. Fine. So guys, this is a serious condition, right? Here, so in the blood and urine of this patient, there is increased amount of uroporphyrinogen 1. Okay, and one more thing that I will take later, coproporphyrinogen type 1. Fine, this is a MCQ, this question has been asked. Okay, then this is congenital. See, the only condition where you will find congenital word is this in the porphyrias, and only condition where you will find acute, I told you. So there is one only acute, there is one only congenital. Okay, and this is the autosomal recessive disease. Fine. So guys, this is also a confusion in a lot of students. Later on you will find this, okay, that some books are mentioning different things. This is the only condition that you have to remember, autosomal recessive. Okay, rest all are autosomal dominant. Okay, right? Autosomal dominant runs in families, each generation, but not recessive. Skip generations are there. Okay, then this child, okay, this baby present with severe sun sensitivity. You know why? Because uroporphyrins, okay, uroporphyrins absorbs UV rays. Okay, so this UP1 accumulate in the skin, right? So this uroporphyrins, these are accumulated in the skin and causes the skin damage. So as soon as the child is exposed to the sunlight, they will be severe sun sensitivity, like they will be like as seen in the movies, they will be what do you say, like as soon as the baby is exposed, there is reddening of the skin and appearance of some bullous lesions, right? Okay, fluid filled. I'll show you the figure. So like that. So that is your severe sun sensitivity. Clear, guys? And later on, the baby may present with a condition known as erythrodontia, right? What do you call it? Erythrodontia means, just write, means the red or the pink colored teeth. Okay, so the first teeth will be red or pink colored. Okay, you can see like this, right? Erythrodontia. It is one of the diagnostic feature, but it appears obviously late. Sun sensitivity is very early, but that may be masked if the patient is at a environmental condition where sunlight is very poor. Okay, cold countries. Fine. So it's like that. Clear to all of you? Fine, guys. So that's called as erythrodontia. Okay. See, guys, the skin. This is the adult, not the child, but this is the skin lesion. Okay. This is the skin lesion that you will find. Okay. Fine. Guys, what happens is this is a case of porphyria in a child, right? Okay, so now moving on to the, guys, previously the porphyria was called vampire's disease, as the vampires are sensitive to the sunlight, just like that. They cannot appear in the sun. As soon as they appear in the sun, they started burning. So that's what you call as vampire's disease. And they have this sun sensitivity and other features like that. Okay, look at the teeth. They later on becomes like the vampires only. Okay, and the body has lots of hairs. That's what you call as hypertrichosis, which is not shown in the movies. I mean to say, like if you see in the movies, the vampires are actually very good looking. Like Robert Pattinson, isn't it? Right? So Robert Pattinson kind of vampires. Actually that's all in the movies, but okay. And now the vampire has become Batman, as Robert Pattinson. Okay. So who's going to watch vampires as Batman? And the bats are vampires, it's already known. Bats can easily become vampires, but now the reverse is happening. Vampires are becoming bats. Okay. Fine. So next is done. Okay, so after that, number 5, step number 5 done, everyone. Number 5 is yes. Why coproporphyrinogen? I'll tell you. It's type 1. Please write that. And I told you it's type 1. Okay, coproporphyrinogen type 1. So the pathway will go from the 1, that's why type 1. Okay. Number 5. So guys, we have now uroporphyrinogen type 3. This is acted upon by the uroporphyrinogen type 3 decarboxylase. Okay, UP3 decarboxylase. Yes, everyone, I think Pushpa Kumar, A. Pushpa Kumar. Are you a DC fan? Yes, I am fan of DC and AC also. Okay, leave it. UP3 decarboxylase. Okay, that's the next enzyme. That's the next enzyme. And what we get next is known as the, so uro means urine, and now copro means the feces, right? Okay, or the stools. So coproporphyrinogen type 3. Okay, type 3. So this is what we get, copro. So where the meaning of copro is stools. So that means it is not water soluble and it will be only present in the stools if in any disease, for example. Like first of all, deficiency of this enzyme, this is called as porphyria cutanea. Okay, porphyria cutanea tarda, PCT. Okay, PCT, right? Who are these guys who are just counting pages only and not listening to the class? How many pages? I have seen a lot of time asking only pages. Okay, porphyria cutanea tarda. Fine. So this is the condition which is seen in adults, and this is one of the most common. This is the most common type of porphyria in the world, right? Most common type of porphyria in the world. Clear, guys? So porphyria cutanea tarda, and these are the patients. Again, they present with the sun sensitivity. Fine. They also present with sun sensitivity, and you can find these patients. Suppose like at the traffic lights, beggars having multiple lesions on their arms and the neck, all the sun exposed area, right? They have this bullous lesions, and they are like beggars, and so they are using this disease to their advantage of like bagging, for example. Okay, so they are mostly the patients of PCT, porphyria cutanea tarda. Okay, sometime you can see, next time if you see such patient, you can make your diagnosis, right? Not always, but yes, very common if such patient you find. Okay guys, so that is one of the most common porphyria. Then moving on to the next step, number 6. So we have CP3, coproporphyrinogen type 3, okay, which is acted upon by coproporphyrinogen oxidase. Okay, coproporphyrinogen oxidase. So what we get is known as protoporphyrinogen 9, right? Protoporphyrinogen 9, and deficiency of this enzyme CP oxidase, this is called as hereditary coproporphyria. Okay, hereditary coproporphyria. Fine. Just the name right? You write the name, not much important because it is very difficult to diagnose, and you have to take the fecal sample, to look for. I'll tell you some more points later. So 6 point, okay, number 6 step. So protoporphyrinogen 9, in short like PP9, right? Take, so that's our next step, PP9, okay, is acted upon by protoporphyrinogen 9 oxidase. Okay, so what we get is known as protoporphyrinogen. Okay, protoporphyrin, okay, type 9. So previously it was a precursor, nogen, protoporphyrinogen, now protoporphyrin, okay. And deficiency of this enzyme, protoporphyrinogen 9 oxidase, fine, this leads to a condition that's called as variegate porphyria, okay, variegate porphyria, clear? Okay, 7 steps, right? And this is the 7th step, and that is what you get, variegate porphyria. Fine guys, and the last step, guys, remember one thing. If a patient presents with acute clinical features, okay, so if there is an acute porphyria, fine. See, I'll show you one thing, and that is, okay, so see, look at this. See, not just basic point I want to tell you, okay, we'll not go into much more details, okay. See, the point here is, you can see, if your patient has acute symptoms, fine, like abdominal pain and neurological, right, then you can simply look for PBG in the urine, right? So if it is not increased, it's not a porphyria. If it is increased, fine, okay, so you will see that porphyrins in the feces are increased or not, right? If they are increased in feces, if they are not increased in feces, then it is acute independent porphyria that I told you, okay. But if they are increased in feces, right, fine, so there is increased amount of porphyrins in the feces, then it can be of two types, right? Variegate porphyria and hereditary coproporphyria, right? Two names, variegate porphyria and hereditary coproporphyria. Depending on, like in the variegate it is protoporphyrin, and in the hereditary coproporphyria the name itself suggests is coproporphyrins, right guys? Okay, so that will be your okay, basic of answering MCQs, fine. So based on this you can easily answer the MCQ, right? So till now questions are not very much common on these other porphyrias except the ones I told you, fine. So acute porphyrias can also be, I told you, the variegate and hereditary coproporphyria. Please write. No need. Okay, acute porphyrias can also be these two, right, with porphyrins in the feces or the stools. Okay, so and acute porphyria can be acute intermittent porphyria only, where the porphyrins in the urine only, right, urine only. So acute porphyrias can be of two. Okay, just no need of that part. Look at this and write this. Okay, acute porphyria, if you get a question, acute means abdominal pain, hallucination, etc. Okay, so how will you answer if it is mentioned that there is a high amount of porphyrins in the feces and that was coproporphyrin in the feces? So you will mark hereditary coproporphyria. Okay, and if it is protoporphyrin in the feces, then you will mark variegate porphyria. Okay, they may mention PBG is also present in urine, it's okay, but in acute intermittent porphyria there is nothing is present in feces. Okay, only urine sample has high amount of. Okay, so that's your you got the point. So that is acute porphyrias. Okay guys, will you able to answer this easily now? Fine, everyone. So last step. Now what is the last step? We are discussing 8. So done. Protoporphyrin 9 that we had is now acted by enzyme which adds the iron. So guys, in the last we add the iron. So that is the name of the enzyme, ferrochelatase. Ferro, ferro, not Ferrero. Okay, ferrochelatase. Okay, fine. So protoporphyrin 9 is converted into heme. Am I clear? Heme. Fine. Done. So this is enzyme which is also inhibited by, so this is also inhibited in the lead poisoning. Okay, in the lead poisoning. Clear guys? So that's your heme metabolism, right? And deficiency of ferrochelatase. Okay, deficiency of ferrochelatase, and this is known as erythropoietic porphyria, right? What do you call this? Erythropoietic protoporphyria. Guys, 8 steps, 8 names of disease, right? Few are very important that I explained you in details. Fine. This is, and if somebody asks you what is a hepatic porphyria and what is the erythropoietic porphyria, so something which has a name erythro is affecting the RBCs or the bone marrow. Okay, and if it is not mentioned erythro, then obviously the disease is affecting mainly liver. Are you getting the point? Okay, So basically speaking, porphyria are of two types. Okay, so one is called as erythropoietic, okay, so one was congenital erythropoietic and this is the erythropoietic protoporphyria. See the name, protoporphyria. And that was congenital erythropoietic porphyria. And the second type is hepatic porphyria. Hepatic porphyria. Okay, so these are the two, and rest all are hepatic type of porphyria. Clear, guys? Hepatic porphyrias. Fine. And please just write the name. Just write the name of this. No need of writing much details, or you can take the photo of this. There is a test, okay, which is called as Watson and Schwartz test. It is again a simple laboratory test, okay, which is also known as aldehyde test. What is the name? Aldehyde test. Okay, so it is used to confirm the diagnosis of acute intermittent porphyria. I told you, you can do one simple thing, that is the urine analysis by the spectrophotometry, and that gives Sauret band. Okay, that gives the Sauret band. Fine, but this is one more test. We can confirm the diagnosis of acute intermittent porphyrias or the porphyrias. Fine, first write this, I'll explain it again. Watson and Schwartz test, just name, please write the name. Okay, Watson and Schwartz test, or it is also known as Ehrlich's aldehyde test, or Ehrlich's aldehyde test is used to confirm the diagnosis of porphyria. That's it. Okay, any porphyria you can confirm the diagnosis of. Clear? At least you should know the names, because these are the common tests performed in the hospitals, right? Okay, on the samples received of suspected porphyrias. Okay, so looking at the new patterns of exams, you should know what is happening around you in the hospitals. Okay, apart from the basic theoretical knowledge, now you should have more of also the practical knowledge that is actually happening around. Okay, somebody was asking me to repeat two types of porphyria. See, porphyrias are two types. If you have the name erythro, it's affecting the bone marrow. If you don't have any particular name like erythro, they are all hepatic porphyrias affecting the liver. I told you, site of heme synthesis only two: erythro, that is bone marrow, and the liver. There are just two sites, bone marrow and liver, right? Okay, what was the test for this name? This what I told you. See, I told you absorbs light. Okay, so what absorbs light? Which instrument? What is the test? Okay, this is on spectrophotometry. Guys, in your first year you saw a instrument known as colorimeter, right? This is the advanced version of the same thing, and this is called as spectrophotometer. Okay, that is used in the hospital labs, right? Spectrophotometer. You got the point? In the first year you did colorimetry experiments. Okay, if you remember. Definitely you remember, right? Okay, so guys, that's your heme synthesis. Heme synthesis. Okay guys, I'm changing the page. And after the heme synthesis, we can take some basic points on heme degradation. Okay, heme degradation. So guys, what is the end product of heme degradation? Answer is bilirubin, right? Answer is bilirubin. Okay, so basically, this point will be covered in your physiology also. So I am just taking some important points here, right? Because we are taking heme. So guys, heme is mostly derived from hemoglobin, right? Okay, I told you 85% of heme is made by the bone marrow, so RBCs and hemoglobin. Okay, so that is your heme. Okay, so this heme is acted upon by the enzyme known as heme oxygenase, right? Heme oxygenase. And guys, this is the only enzyme in our body which produces us carbon monoxide. Okay, carbon monoxide. Okay, let me write this so that you can add it to your notes. Okay, see, so just write the heading heme degradation. Okay, heme degradation. So in the heme degradation, so first of all, it can be hemoglobin or any other heme proteins, right? For example, CYP, which gives the free heme. Okay, this free heme is acted upon by the enzyme that's called as heme oxygenase. Guys, heme oxygenase is something which breaks the the bonds. Okay, methane bridges. See this. Look at this. Okay, so this is the structure of the heme that I told you. Fine, and these are linked by these four rings are linked by some bridges. Okay, they are linked by some bridges. Okay, like this. Okay, suppose this is the one bridge, this is the bridge. Okay, so these bridges are broken and the CO is released, not CO2 but CO, carbon monoxide. Okay, so we are breaking the heme, which is four rings. Okay, fine. So heme oxygenase, yes, you have to write this, please write heme oxygenase, and this is releasing the carbon monoxide. Okay, and this is also releasing the iron, right? Fe3 plus. Okay, so guys, the only enzyme which is releasing carbon monoxide in human body is this one, right? That is heme oxygenase. Okay, heme oxygenase. Okay guys, and this produces the biliverdin, and then we take the enzyme that's called as biliverdin reductase. Okay, biliverdin reductase. Okay, and this biliverdin reductase will generate the bilirubin. Okay, will generate the bilirubin. Okay guys, and this uses NADPH. So what are the enzymes which generate NADPH? You should know, right? You must be knowing already, right? What is the major source of NADPH? Any answers from you guys? What are the sources of NADPH? This is the use of NADPH. Fine. So what we get is bilirubin. Okay. Now is this bilirubin, okay, is transported to the liver. Fine. Where it is conjugated. So it is first transported to the liver where it is conjugated, and then it is excreted, okay, in the bile, right? So it is conjugated okay, conjugated in the liver cells, right, by the enzyme that is called as UDP glucuronide transferase. Okay, UDP glucuronide transferase. Okay, so guys, this bilirubin, I'm continuing. So this is what happens, the heme degradation, and heme degradation took place in various tissues which are known as reticuloendothelial cells, like spleen. Okay, for example, spleen. Okay, so this is the degradation occurs and bilirubin is made. Now this bilirubin is released to the blood, right? Okay, fine guys. Yes, HMP is the right answer. Very good. Okay, pentose phosphate pathway or HMP, right? So blood, this bilirubin is insoluble. Fine. So in the blood, albumin binds the bilirubin and transport. So guys, what is the function of albumin? See, these are the basic things that we should always understand and remember. Okay, like albumin, like simplest of thing, what are the functions of albumin? You should know. It's a transport protein. What it transports bilirubin, it transports drugs, it transports fatty acid, it transports hormones. Which hormones? Like aldosterone, fatty acids, drugs. Okay, so like fine. So bilirubin is one of the important. Calcium is very, very important thing transported by the albumin. Okay, so albumin binds the bilirubin and transport to the liver cells. Okay, transport to the liver cell. So in the liver cell, right? So suppose this is the liver cell. Okay, like this. This is the liver cell, and the albumin has been bilirubin has been given here. Now this bilirubin binds ligandin, okay, or also known as Y protein. Okay, or also known as glutathione S transferase. Fine. So this binds to this GST. Okay, glutathione S transferase. Previous names were ligandin and Y protein, which are not used nowadays. So the new name is glutathione S transferase. So what is the first thing in the liver which binds the bilirubin? Okay, we don't want bilirubin in blood, right? We want bilirubin to make soluble and excrete it in the bile, right? This is what we do, and this will be entering into the intestine. Okay, so we want to make it soluble. High bilirubin should not be there in the blood, right? It should not be beyond 1.2 mg per dl. That is the amount of bilirubin we have, total bilirubin in our blood. Okay, so glutathione S transferase is the name of the enzyme. Fine guys, so this glutathione S transferase So this GST binds the bilirubin and transport to smooth endoplasmic reticulum. Okay, so this all happened in cytoplasm. So this is a liver cell cytoplasm, and now we have transferred it to the smooth endoplasmic reticulum. Okay, so suppose now let's say this is the smooth endoplasmic reticulum. Okay, so in the smooth endoplasmic reticulum, this is the bilirubin, and guys, bilirubin is conjugated, okay, by the addition of UDP glucuronic acid. UDP glucuronic acid is added here, and this is called as conjugated bilirubin, which is soluble. Okay, conjugated bilirubin, which is soluble. So guys, this enzyme is called as UDP glucuronide, okay, transferase, right? UDP glucuronic acid transferase. Am I clear, everyone? Okay, UDP glucuronic acid transferase. So I made the bilirubin water soluble, right? So now this is soluble, and this will be excreted by the liver to the bile, right? Through the bile, right? Okay, so the bilirubin has been conjugated, and now this bilirubin will be delivered with the help of a protein that's called as MRP2. Okay, please write the full form. Multi-drug resistance protein. Please write the full form of this. Multi-drug resistance protein, which is also known as CMOAT. CMOAT. What is the full form? Canalicular. Please write, because we are transferring this to bile canaliculi. Okay, from the smooth endoplasmic reticulum of liver cell, now we will transfer this to canalicular. C for canalicular, M for membrane, organic anion transporter, right? Organic anion transporter. Clear? So guys, this will be transported to, let's see, just to understand that this is the bile canaliculi. Okay, bile, fine, and this is MRP2. Okay, so like this. Fine guys, so conjugated bilirubin will enter here. Okay, very simple, very clear, very important. And you know the disorders that you will read later on, probably in pediatrics, I'm just writing the names that you can correlate here easily. And the deficiency of okay, UDP, okay, glucuronyl transferase enzyme, right? This leads to unconjugated hyperbilirubinemia. So bilirubin levels will be high, but which one? Unconjugated. Okay. And one of the most serious of all is Crigler-Nazar syndrome. Okay, that you will read more in your pediatric lessons. Okay, Crigler-Najjar syndrome. I told you the name of enzyme, which is very, very important here. Okay, so Crigler-Najjar syndrome. And second point, if there is deficiency of CMO8 or MRP2, so obviously the bilirubin is conjugated, see, but it is not able to excrete it, right? So there will be conjugated hyperbilirubinemia, right? Conjugated hyperbilirubinemia, and this will be, for example, the most serious one is Dubin-Johnson syndrome. Okay, right, guys. So this is the basics of heme metabolism that you must understand and remember. Okay, the total bilirubin is around 1.2 mg per dl in our blood, right? Okay, and most of it is the unconjugated one, but it is very less, very, very less amount. Okay, very, very less amount. Fine, guys. Okay, recently this year in all India exam there was a MCQ, and that MCQ was based on the analysis of bilirubin. Okay, and that analysis of bilirubin was based on a test that we call as Vandenberg reaction. Okay, so it's a very simple test. Just you should understand the name. Okay, Vandenberg test. Okay, Vandenberg test is basically, okay, it's a very first of all test which is very much details are there. Okay, so Vandenberg test is something which is detecting the bilirubin, okay, in the blood, and this gives us the values of two things. One is called as direct bilirubin and other is called as indirect bilirubin. Okay, so when I'm saying total bilirubin, it is made of two things, okay, conjugated and unconjugated. So T-bill is 1.2 mg per dl. So when you look at the reports in lab, right, the labs gives you two results only, T-bill, D-bill. Okay, D-bill should be 0 to 0.2 mg per dl. Now what is D-bill? What is T-bill? See, guys, what I mean to say, this is your lab report of a patient, your patient, and the patient is, let's say, suffering from jaundice. But this is a normal report. So where the T for total bilirubin and D for the direct bilirubin. When I say direct, the word direct came from this test that I'm talking about, Vandenberg test. Okay, so D for direct, and direct means conjugated. So that means most of the bilirubin present in our blood is unconjugated. Obviously, once it is conjugated, it should not come back in the blood now. Are you getting a point? Once your this bilirubin has been conjugated, it should not reach to the blood again, right? Obviously. So the direct bilirubin or the conjugated bilirubin level should be very low in the blood. Fine. If it is high, that is diagnostic of some disorders like some types of jaundice. Okay, fine. So I'm just writing a test which was recently asked, Vandenberg test. Okay, so this is the principle that is used in the laboratory. Okay, so VDB in short, So Vandenberg test, can identify the type of, okay, we can identify the type of jaundice, okay, type of jaundice or bilirubin. Bilirubin means whether it is conjugated or unconjugated, right? Fine. So Vandenberg reagent, no need to remember the name, right? There is a Vandenberg reagent. So Vandenberg reagent react with conjugated bilirubin, fine, and this gives purple color in few seconds, okay, fine. And this is known as direct bilirubin. Understood now, please? Okay, if you understand this, it will be very easy for you to understand a lot of topics related to jaundice, testing, bilirubins, hyperbilirubinemias. Okay, this makes your basics. Fine. So Vandenberg reagent, there is some reagent. It reacts with conjugated bilirubin in the serum, in the blood. Okay, sample of the patient that you have. Okay, and it will react instantly, within seconds, and will give the color. So that is a direct bilirubin. Okay, fine. Acha, see, before starting this, one more thing. Vandenberg reagents, okay, will give no color in the normal blood, because I told you now, in a normal blood we have very low or almost zero direct bilirubin. Okay, reacts with conjugated bilirubin, right? This is your patient sample, right? Okay, Vandenberg test gives no result in normal blood. First you should understand this, because there is no direct bilirubin. Okay. Now the second point. Okay, so when you take the Vandenberg reagent, okay, plus then you take patient sample, right? Patient has jaundice already. Okay, this patient also had jaundice, suppose. Okay, then this gives no color, suppose. But why? There should be some color now. Okay, but it may be unconjugated hyperbilirubinemia, right? And unconjugated is not water soluble. So what we will do, we will add methanol, okay, which solubilize unconjugated bilirubin. Please write. Okay, now we are testing unconjugated indirect bilirubin. So you have the sample of the patient who had jaundice, but you don't know what is the cause of jaundice, right? So you took the VDB, you took the sample, but there is no color. You add the methanol, which solubilizes the unconjugated bilirubin, right? Okay. Now, so this will take around 30 minutes, not important for you. Now the purple color appears. Now the purple color appears, and this is known as indirect bilirubin. Am I clear? Indirect bilirubin. Fine. Okay. So that is unconjugated bilirubin. Clear, guys? Okay. So that is how you can understand the blood report easily. Okay. Suppose the total bilirubin was 5. In your blood report, the patient bilirubin T bill was 5, but D bill was only 0.2. That was normal. Okay. So that means it is a high rise in unconjugated bilirubin. Okay. Fine, guys. Okay, we'll come to that point. Now the third point is biphasic reaction. Okay, Vandenberg test. Biphasic reaction. No, see, normal blood will give no color, because I told you now, the bilirubin levels are very low in the blood normally. Now it is 1.2 is the maximum limit. Okay, the T bill may be less than that also. So I mean to say it is almost negligible amount of bilirubin present in our blood. Obviously, like if it is high, then your sclera will be yellow. Okay, so there is negligible amount of bilirubin and it will give almost no color. Okay, even after adding methanol, it will give almost no color. Fine. Okay. Right, guys? Okay. So, indirect bilirubin. Now, biphasic reaction, Vandenberg test. Okay, now we'll classify the jaundice also. First, see this biphasic reaction, Vandenberg test. Okay, biphasic. So, you have again the reagent. Okay, you have the reagent plus you have the patient sample, right? Blood sample. Then it gives purple color. Okay, purple color. So that is conjugated bill is present. Fine. Then you add methanol. Okay. After 30 minutes of the reaction, the purple color intensifies. Okay, the purple color intensifies. Fine. And this is your indirect bilirubin. Indirect bill or the unconjugated bilirubin. Okay, unconjugated bilirubin. So both the type of bilirubin are raised. So guys, which condition both the type of bilirubin are raised? So I mean to say, see, the jaundice, that's not my topic, but now you will have very clear concepts of jaundice when you read somewhere, maybe in your pediatrics or physiology or medicine or wherever, right? So jaundice, there are 3 types of jaundice. Okay, so first is called as prehepatic. So before reaching the liver, prehepatic. Okay, so prehepatic jaundice, for example, one of the most common type of prehepatic jaundice is hemolytic. Okay, hemolytic, yeah, hemolysis. Right, guys, so you are done with your G6PD deficiency. Yes, it's a qualitative test, right? not a quantitative, it's a qualitative test. Okay. Fine. So hemolysis. So guys, hemolysis, like in G6PD deficient cases or in microbiology, you must have a lot of infectious cases, right? So hemolysis, okay. So there is increase in unconjugated bilirubin or the indirect bilirubin. So guys, that is called as Vanden. This is increase in unconjugated bilirubin. So indirect test. So what do you call this? Indirect test. See, we can write like this. Okay. Okay, just a minute. Okay, we can use these words. These are known as, please write this, indirect positive, direct positive, and biphasic that I mentioned. Okay. So what I told you, increase in unconjugated bilirubin. So what is the Vandenberg test will be? This is Vandenberg test is indirect positive. Okay. Now second type of jaundice that you know is the hepatic. Okay. Hepatic jaundice occurs in which case? Like hepatitis B or any condition where the liver failure. Okay. Or the liver failure. Right. So guys, in this case actually there is increase in both type of increase in conjugated bilirubin plus increase in unconjugated bilirubin, both occurs. Okay. In the hepatic jaundice, both the type of bilirubin will be high. So this is the biphasic reaction. Actually liver has less capacity for both the things. Liver has less capacity to conjugate the bilirubin, so that's why unconjugated rises, and liver has less capacity to secrete the conjugated bilirubin, so conjugated also rises. Got the point? Liver, in this case, liver fails, so liver has less capacity to conjugate also, and to, after conjugation, to excrete also, because both are demanding, energy demanding processes which liver cannot do. That's why both rises. Okay. And number three is post hepatic jaundice. Okay. Post hepatic jaundice is also known as obstructive jaundice, and in this case guys, this will be direct direct positive Vandenberg test, because there is increase in conjugated bilirubin. So what is the example of obstruction? Like the cancer of heads of pancreas, which is obstructing the bile duct, pressure on the bile duct. Am I clear? So that's all. Based on this, you can understand any topic related to bilirubin and jaundice and everything. All the syndromes that I mentioned will be very easier after understanding this. Okay, pre-hepatic, post-hepatic, hepatic, right? So that's all. This was a recent MCQ. Vandenberg is a qualitative test, I told you. Okay guys, now we'll start with our next topic, and that is nucleotides. Then I just want to show you one. Okay, I'll show you the question later on. Okay. So next topic that we'll take is nucleotides. So heme is covered, amino acids are covered. So your metabolism is almost over except this last one. Okay. And with the nucleotides I will start with the genetic section then. Fine. Okay. Then guys, so that's your, so let's start with the nucleotide metabolism. Guys, nucleotides, as you know, like, you know, these are the things that you know from your school times and everything, right? Even, you know, glycolysis and everything from your school time, right? We are going one more step ahead, that is understanding in a better way, and, you know, understanding that disease and disorders and applications in the medical field, right? Okay, that can be asked as MCQ. Yes, I will teach everything. Enzymes also I will teach. Okay, enzymes are very simple. Okay, nothing important, much difficult in enzymes. Okay, guys. So nucleotides are. So first I'm taking purine nucleotides. Okay, purine nucleotides. Acha guys, bilirubin normally doesn't appear in the urine. Okay, there was a question asked to me yesterday by some student. Okay, see, bilirubin normally doesn't appear in the urine. You know why? Because it is bound to albumin. If it is present in blood, it is bound to albumin, so it will never appear in urine. Okay, but in case of conjugated hyperbilirubinemia, which is a water soluble type of bilirubin, right? It can appear in urine. It is there actually, and it will be absent in stools. Okay, because it is not secreted. Now, conjugated hyperbilirubinemia means obstruction, so it will not appear in the stools. Are you getting a point? So stools will be clay colored without any pigment, but urine will be dark colored with bilirubin as a pigment, very dark colored, right? So bilirubin, yesterday somebody asked me this question. So bilirubin doesn't appear in the urine normally because it is bound to albumin. Okay, but if it is direct bilirubin, which is normally zero in blood but increase in the conditions of conjugated hyperbilirubinemia, right, like the obstructive jaundice, gallstones, so then it may appear. Clear to all? Okay guys, purine nucleotides. So nucleotide is nothing, first of all, you know, nucleotide is sugar plus base plus the phosphate that you already know. So what are the examples of the purine bases? The purine bases that we have, as you all know already, are adenine, guanine, hypoxanthine, but which is not found normally in the DNA. These two only found in the RNA and DNA, right? Okay. So purine bases, adenine, guanine, hypoxanthine. Yes, for MCQ go for DQB, and if you find any discrepancy in the answers, please follow notes first. Sometimes there may be some discrepancy in the answers, may not be. Okay, then you follow my or Dr. Poonam's notes only first for the answers, and then you can confirm with us. Purine bases. So guys, purine bases are adenine and guanine, right? So structure, general structure that you should know, it is a ring structure. Okay, so ring structure, right? with the 9 atoms. Fine. So they are numbered like this: 1, 2, 3, 4, 5, 6. Okay, so first string is like in the anti-clockwise direction. Okay, and 7, 8, 9. There are 4 nitrogens. Okay. So these are the 4 nitrogens. Clear? Now the first important question is, source of the nitrogen and carbons in the purine ring. So guys, the N1 is from aspartate, okay, N3 and N9 from the glutamine, carbon 4, carbon 5 and nitrogen 7. This is from glycine. So this structure, see here, 4, 5 and 7. Okay, like this, 4, 5, 7. Right. Okay. So these are the sources of nitrogens total. So aspartate, glutamine and glycine, 3 amino acids are involved. Very important. These are the 3 amino acids. Okay, C2 and C8, they are derived from folate. Very important. Carbon number 6 is from CO2. Right guys? So in the purine metabolism, this one is the most repeated question, right? That sources. Okay, right, so that is your sources. This is the basic biochemistry. Okay, you should know the sources of synthesis. Now synthetic pathway is very, very long and we will take some important steps. So now write the heading: purine nucleotide synthesis. Okay, purine nucleotide synthesis. No, 4 and 5 are glycine. Purine nucleotide synthesis will start now. So first of all, nucleotide we have to make. So I want sugar plus phosphate and then I will make the base. So first step, we will take ribose 5-phosphate. Anyone, what is the source of ribose 5-phosphate? So ribose 5-phosphate, which is taken from the HMP shunt. Okay, it binds with ATP and it generates a structure which is called as, so this is a pentose sugar. Okay, this is a pentose sugar. And this is called as phosphoribosyl pyrophosphate. Okay, this is what you called phosphoribosyl pyrophosphate, in short PRPP. Guys, this is your, for every purpose you will count it as sugar plus phosphate. Fine. So what we made, phosphoribosyl pyrophosphate. So 3 phosphates are here total. Clear? And guys, this enzyme is called as PRPP synthetase. Okay, PRPP synthetase. Right. Overactivity. This is the only enzyme in biochemistry that probably I'm telling you, right, where I'm talking about overactivity, right, not the deficiency. So overactivity of the PRPP synthetase. Okay, PRPP synthetase, yes, you all are correct, HM patient. So overactivity of PRPP synthetase leads to hyperuricemia or the gout. I'll tell you why. Because of excessive amount of the purine nucleotide synthesis. But you know why I'm writing here, but you will not be able to understand right now. I'm just writing so that we can later on understand it. PRPP is the stimulator of the rate limiting steps of the purine synthesis. That's why. Okay. I'm sorry. Fine. So rate limiting step of purine synthesis, overactivity. Right guys, so let's see next step, number 2. So now we take the sugar plus phosphate and now we'll make the base. So now there are number of steps, but I'll take some important ones. Glutamine is added here, first amino acid, and this enzyme is called as PRPP glutamine. Okay, PRPP glutamine amido transferase. Okay, PRPP glutamine amido transferase. Right. So kuch nahi yaar, we will just add the nitrogenous compounds on the sugar. So what we get is phosphoribosyl and amine. Okay, phosphoribosyl pyrophosphate was there, so we removed the pyrophosphate and we get the amine. So phosphoribosyl amine, that's it guys. This is the very important enzyme, rate limiting enzyme of this pathway. This is regulation, which is very simple to remember. I told you already, high availability of sugar plus phosphate activates this enzyme. Now you can correlate here that why there was hyperuricemia in the von Gerke's disease. That must be already covered, right? You know why? Because in the von Gerke's disease, glucose 6 phosphatase was absent, so G6P increases. G6P enters via HMP shunt, G6PD, so it enters HMP shunt, makes lots of sugar. These sugars makes PRPP ultimately here, and this PRPP activates purine nucleotide synthesis. So the sugar G6P, which was in the von Gerke's disease, type 1 GSD, is activating purine nucleotide synthesis like this. You got the point? This is sugar plus phosphate. So, and I told you why the PRPP overactivity can also lead to hyperuricemia. Okay guys, and it is inhibited by the end products AMP, GMP, IMP. IMP is inosine monophosphate. So these are inhibitors, allosteric inhibitors of this enzyme. Okay, Very important to understand various concepts. Okay, and you should know the regulation part, right? Did you all? Now this phosphoribosylamine will undergo number of steps, and this will make a compound that's called as IMP, inosine monophosphate. Okay, that's called as IMP, inosine monophosphate. This is the parent. This is called like this, parent purine nucleotide. It is not found directly in the any DNA or RNA. What is I? I here. I for inosine. I is made of the base. Okay, so it is a nucleoside, hypoxanthine plus sugar. That's called as I. I for inosine. Okay, guys. Now this IMP. Next step. This IMP is converted into first of all, let's say GMP. This uses ATP. Okay, fine. And the name of enzyme is called as IMP dehydrogenase, and two enzymes, GMP synthetase. Okay, so see guys, okay, first write, then I'll tell you some important points. Okay, and then this IMP is converted to AMP. Okay, so here we need GTP, right? So GTP is required here. Clear? So this enzyme is called as adenylosuccinate synthetase, and same, okay, and adenylosuccinate lyase. Okay, adenylosuccinate synthetase and adenylosuccinate lyase. Now what I want to tell you is that this will ensure equal amount of A and G production. If I use, for making A, I am using G nucleotides as energy, and for making G, I am using A nucleotides as energy. Okay, so this will balance the amount of A and G produced in 1:1, because obviously I want the equal concentration of A and G before I start making DNA or RNA. It should not be like I have high amount of A, low amount of G, like that. Okay, otherwise this will lead to incomplete replication of synthesis of DNA. Clear guys? So please highlight this point. Okay, for making A, I need G nucleotides. For making G nucleotides, we need ATP as a source of energy, right? And okay, so using GTP for AMP synthesis, okay, and ATP for GMP synthesis ensures 1:1 ratio of A and G nucleotides, okay, in cytoplasm. I'm to say, yeah, this is made initially in the cytoplasm, okay, then they will be used in the nucleus later on, right? In cell we can write. Okay, fine guys, everyone. So that's your basic point. And one drug that you will read or you must be knowing already, mycophenolic acid, right, which is used to prevent, okay, used to prevent the graft rejection, okay, used to prevent graft rejection, is inhibitor of IMP dehydrogenase. Okay, IMP dehydrogenase. Clear guys? Okay, IMP dehydrogenase, inosine monophosphate dehydrogenase, right? Okay, so now the next point is, so we get AMP and GMP, but we cannot use them for making the DNA. Why? Because they are not deoxynucleotides. For making DNA, I want deoxynucleotide, so I have to convert AMP into DAMP and GMP into DGMP. So for that we need a separate enzyme. So what is the next step? So deoxynucleotide synthesis. Okay, so first of all, AMP and GMP, right, by the various kinases present in our cell are converted into GDP and ADP. Okay, so let's say this is the first step. Fine. And some will be directly, okay, so ADP and GDP. Now these diphosphate nucleotides, right? So I use ADP and convert this into deoxy ADP, right? Or I use GDP and I get deoxy GDP, like this. How? So first we will write and then we'll see. So what is the name of enzyme? Not simultaneously. First we'll make Nisarg Patel. First we'll use AS synthetase, adenylosuccinate synthetase, then adenylosuccinate lyase, obviously. Similarly, there IMP dehydrogenase first and then GMP synthetase. The point is not unnecessarily burdening of the reactions, right? Wherever possible we can omit the unnecessary reactions. So that's what I did. Okay, so the names are not important here except IMP dehydrogenase because that's a target of drug. That's it. Okay, so diphosphate nucleotides पे हम ये करेंगे अब, है ना? We will not do it at the level of this. You know why? Because monophosphate derivative वाला कोई enzyme नहीं है. See, the name of enzyme is, what is the name of enzyme is? It is nucleotide diphosphate reductase. Okay, nucleotide diphosphate reductase. Okay, so we are reducing. Okay, and guys, this needs a coenzyme, and what is the coenzyme here is thioredoxin is required here. Okay, thioredoxin will be used here, right guys? So we also call this ribonucleotide diphosphate reductase or nucleotide. We can also write like this, ribonucleotide nucleotide diphosphate reductase. Okay, so basically यार हमने क्या किया, we did nothing but we just use the sugar for deoxygenation, as you know. Now understand this concept. See guys, this is sugar, as you all know. Sugars, this is the oxygen of the CHO. You must be knowing already, you're done with your carbs. Okay, this is the 5 carbon sugar. This is called as 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Okay, so the numbers of carbons are named like this: primes, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Primes, just like when I say DNA is in 5 prime to 3 prime direction, actually I am talking about the direction of sugar, as you know, right? And then sugar has OH groups, right? As you all know, what are carbs? Carbs are alcohols. Okay, aldehydes and keto derivatives, alcohols, right? So we all are alcoholics because carbs make 60% of our diet, isn't it? Okay, leave it. So we have this OH group are present on the pentose sugars, right? So I am just taking second and third, and while deoxygenation, you know what is done. Okay, so we have removed the oxygen from the carbon number 2, right? Oxygen from the carbon number 2. Guys, generally the base binds at carbon number 1. So suppose this is ADP, phosphate, phosphate. Okay, phosphate, phosphate. This is A. So if this was ADP, this is 2 prime deoxy ADP. Okay, everyone. So what is the name of the enzyme which is used here? Reduction. Removal of oxygen is reduction. We have removed this oxygen, right? That's called as reduction. Guys, what happens if you remove this oxygen also? There will be no DNA replication. This is the principle of making drugs which are known as dideoxynucleotides. Antivirals like suppose zidovudine. Zidovudine in anti-HIV therapy. If you remove this also oxygen, they are called as dideoxynucleotides. And if all the oxygen second and third both are removed, especially this third one, then how the DNA will be made in 5 prime to 3 prime direction, because we have to add the phosphate at this position of the incoming nucleotide. Okay, so this is deoxynucleotide is made. That's it. What is the name of the coenzyme? Thyrodoxine. Very important. Okay, MCQ. Coenzyme required for reducing ribonucleotides. Okay, what is the name of enzyme with drug which inhibits this? This is a drug which is not commonly used nowadays, okay, except in certain cases like melanoma. Hydroxyurea. Hydroxyurea is an anti-cancer drug which inhibit this step. Obviously, like this will inhibit DNA synthesis, okay, so that's your this is the drug anti-cancer drug hydroxyurea. Fine, which is also used in sickle cell disease by a different mechanism, right guys? Okay. Now regulation part. Okay, the deoxy ATP, okay, inhibits the ribonucleotide. It inhibits the ribonucleotide diphosphate reductase enzyme and prevent the conversion of GDP to DGDP or you can say anything like CDP to CDP, or whatever else we have, TDP to the deoxy, TDP, etc. So it prevents all the reactions, all the reduction reactions by this enzyme. So this is the most potent inhibitor. Okay, this is the most potent. So I'll tell you the application of this in SCID, severe combined immunodeficiency. That's your one of the disease that you must be knowing in your immunology section. Okay, SCID. So remember this, only one regulation, deoxy ATP. Okay guys. Okay, fine. Now moving on to the next point. So now we'll take a very important point, guys, and that will be, okay, break. No break, yeah, please. We'll take a break. Sure, you want a break? Okay, then yes. If you wish, your wish, my command. No, I can't say that. Okay, so we'll take a small break. Come back in 10. Okay, come back in 15 minutes. Right? We'll start. Mansi Ghorpade, saturated. No, please don't saturate. I'm taking break. Be unsaturated. They are more healthy. This is the purine salvage pathway. Okay. So purine salvage pathway. Okay. Okay. Before that, we can actually understand that what is the end product of. Just change it. First, we'll take purine degradation pathway. Okay. Purine degradation. So, guys, what is the end product of the purine degradation, as you must be knowing all, and that is uric acid. Okay, that is uric acid, the purine degradation product. Okay, and excessive uric acid is not good for health, as you know, right? One of the common disease that can occur is gout. Okay, apart from others like urate nephropathies and affects the joints. It can even causes insulin resistance. There are a lot of things, but first we should understand that purine degradation pathway has one important enzyme that we should know in the name of SCID, disease SCID. So let's start. Suppose I'm starting with AMP. Okay, so I'm taking one example. AMP is a purine nucleotide. Okay, so I'm taking the first enzyme which is present in the bile canaliculi, and that is called as 5' nucleotidase. Guys, purine degradation pathway, the major site are the two, liver and intestine. Okay, so this occurs mostly in the liver or in the intestine. Now when I'm talking about intestine, it is all I'm talking about dietary. Okay, so dietary purines which are present in anything which is having DNA or nucleic acid, all the meats, even sprouts. Okay, even the alcohol, especially the alcohol which are brewed, like the beer especially, which contains high amount of purines. So dietary purine doesn't contribute to the DNA synthesis. You should know this. Okay, you please write this line, everyone, please write this line. Dietary purines, okay, dietary purines do not contribute to the purine synthesis, DNA synthesis. Okay, so they are degraded. Okay, they are mostly degraded, right? Okay, this can be important, right? So otherwise the major site is liver. So this enzyme, as you all know, is present in liver, 5' nucleotidase. So what is the function of this enzyme? This enzyme removes the phosphate. So what we get? We get adenosine, a nucleoside. Then this is acted upon by adenosine deaminase, that is called as ADA. Okay, adenosine deaminase. Okay, then this ADA enzyme, this causes deamination of A. So A is converted into hypoxanthine. So the difference between A and this is this is only amino group. So guys, hypoxanthine containing inosine is formed, right? Okay, inosine is formed. Now the next enzyme is purine nucleoside phosphorylase. Okay, purine nucleoside phosphorylase. And this removes the sugar. So obviously now we are left with hypoxanthine. Okay, hypoxanthine, which is acted upon by the enzyme which is called as xanthine oxidase, XO. This enzyme is molybdenum dependent. Okay, molybdenum dependent. And guys, this enzyme is oxidase, so it uses oxygen and produces H2O2. So people who are overproducers of uric acid, they are actually at risk of reactive oxygen species induced damage to the cells. Are you getting my point? H2O2 damages cell. So it's not the uric acid. Before uric acid, even H2O2 is damaging the cells. So the people who are overproducing uric acid, they are actually at higher risk of damage by H2O2 also. Okay, so free radical generator is produced. Okay, so what we get is xanthine. So this damages cell, remember it. Xanthine, again xanthine oxidase, again H2O2. So guys, what we get is known as uric acid. So guys, uric acid ring is not broken down, ring is still the same. Okay, we have the same ring, just we have 268 position, we have the 3 oxygens, right? 3 oxygens, that's it. Now some of the questions. Question number 1. Okay, SCID, right? What is SCID? Severe combined immunodeficiency. Now guys, this is a disease which has multiple etiologies that actually you will cover in your immunology. This one and its mechanism I'm explaining because probably it's not covered anywhere, right? I will cover also SCID in the DNA repair mechanism, just 1 or 2 points, okay, which are important MCQs. SCID, why? SCID for severe combined immunodeficiency, right? B plus T cell both deficient, as you know, that is called as SCID. So this is due to deficient enzyme ADA, adenosine deaminase, okay. Which enzyme? This deficiency of this leads to SCID, adenosine deaminase, okay. So if it is absent, what will happen? There will be increase in the amount of adenosine. Now there are two reasons which are saying that why this happens, okay. First, this adenosine directly, right, induce apoptosis of B plus T cells, okay. Second, this adenosine convert to deoxy ATP by kinases, okay, by the kinases. And I told you deoxy ATP is the inhibitor of which enzyme? Of the ribonucleotide diphosphate reductase. Please write the full form, I told you already. Ribonucleotide diphosphate reductase, right? Okay. So decrease in deoxynucleotide, and which leads to decrease DNA replication or proliferation of B plus T cells, okay, which need to be proliferated millions or billions of time during infection, okay. So they need to divide at a very fast rate, okay, to kill the microbes or to, you know, trap the antigen, whatever, right? So as you know, this will be your. So guys, these are the two basic reasons, and especially remember this one, okay. DATP will lead to this problem, major problem, and that is called as adenosine deaminase deficiency. Severely immunocompromised child, also known as bubble boy disease, because you know the boy X-linked disease. So boy has to be capped inside a bubble of the, you know, plastic with oxygen supply, so that, you know, the foreign particles, microbes, viruses, fungus present in the air does not enter into the system. So they kept an isolated environment, okay. You got the point, guys? So that's all about the severe combined immunodeficiency. Okay. Second question of this topic is that deficient xanthine oxidase, okay, deficiency of xanthine oxidase will lead to obviously hypouricemia, right? Less than 2 mg per dl. Normal is 2 to 7 mg per dl, okay. So hypouricemia, rarest of rare diseases, right? But MCQs here, so that's the point. And guys, one more point. Deficiency of xanthine oxidase can also lead to a condition known as Xanthinuria or hypoxanthenuria. Actually, it is named as xanthinuria. Fine. Bubble, not bubble ball here, bubble boy. Not the ball, bubble boy disease. Okay. RDR, nothing. Yep, RDR, Shreya, this is ribonucleotide diphosphate reductase, same enzyme I told you, no, which is inhibited by DATP. Same enzyme is writing here. Okay. So xanthinuria. Okay, and hypouricemia. Fine, guys. Okay, so that's your. And gout, guys, gout, like alcohol induces gout. Okay, because alcohol, or not all the alcohols, not the spirits. Okay, alcohols which are rich in purines like yeast. You know, beer, for example, is totally contraindicated in the patient of known gout. Beer. Why? Because it contains high amount of purine nucleotides in the form of this yeast. Okay, yeast is required for the process of brewing, brewer's yeast. Okay, fine. So that's that's more dangerous. And second thing, alcohol also causes dehydration, which can lead to precipitation of uric acid. Okay, you will read more about gout in your other subjects. Fine. So now next point is purine salvage. Okay, pathway. So salvage means to reutilize. Okay. So purine salvage pathway, first of all, the major site of purine salvage pathway is brain. Okay, and WBC. Okay. Why brain and WBC? As see, salvage means I will not waste any purine nucleotide and make uric acid. I will reuse it, recycle. Okay, recycle. So brain, as the, why brain so important? As the adult brain has no purine de novo. De novo means new. De novo synthesis pathway. Okay, so guys, adult brain cannot make nucleotides. Okay. Fine. You got the point? So as the adult brain has no purine de novo synthesis pathway. Clear? So the brain is a major site of salvage. Let's understand salvage. What is salvage? So see, a cell, everyday cell dies, right? Guys, a cell can divide only for 50 to 70 times, and that's what you call as Hayflick limit. Okay, so cell divide only for 50 to 70 times in the life. Okay, this is known as Hayflick. The name has been given by the scientist, Hayflick limit. This was a recent MCQ which actually I thought to cover in telomerase, but okay, I'm writing here 50 to 70 times. Okay, that's called as Hayflick limit. So that is a limit of any cell's life. So then this will lead to apoptosis or death. Okay, this leads to one of the important part of apoptosis is DNA degradation. Right, DNA, when I say DNA, I mean to say both purines and pyrimidines. Okay, right. So both purines and pyrimidines are degraded, right? But I am more concerned about the purines here. You know why? Because we are taking that topic. So DNA degradation or the purine degradation. Okay, let's take first DNA degradation. Free bases A and G, also C and T, but I'm taking only this. Free bases like C, T, but I'm talking about A and G released. Now this free base can form uric acid. Okay, right? This can form the uric acid free base. Okay, in the purine degradation pathway. Fine. So A, G, even hypoxanthine, right? I can take like this, or I can simply use a process that's called as salvage. So in the salvage I will just add sugar plus phosphate. Okay, and what is sugar plus phosphate? PRPP. So that I will add here. And what I get? IMP, GMP and AMP. So guys, nucleotides are again made na? Very simple process. Okay, clear everyone? So IMP, GMP, AMP is made. So these are the nucleotides which are again made, and this will be reused. So the body has saved a lot of energy. Body has saved itself from unnecessary uric acid generation, and brain has only this pathway left. You getting a point? Salvage means to recycle. Okay, salvage means to prevent losses. Okay, so to recycle, to prevent losses. Okay guys, understood everyone? कैसे किया हमने salvage? जो free whatever the free base that was left, just we added the sugar plus phosphate and we get the nucleotides that I can use again. Okay, brain is the major organ because it has no other pathway left. All other body cells can do it, right? Major pathway is that. Okay, now there is a one very important question here, and that is okay, first of all reaction देखते हैं हम. So I am taking hypoxanthine, HX for hypoxanthine, and guanine. Okay, name of enzyme is HGPRT, and what we get is IMP and GMP. I'm not taking A because there is no clinical significance of that. Okay, so hypoxanthine and guanine, name of enzyme is and this we add PRPP. Okay, PRPP, sugar plus phosphate हमने ऐड किया. What is the name of enzyme? Hypoxanthine guanine phosphoribosyl transferase. Please write. No need to even write. H you know, G you know, hypoxanthine guanine. PRT का full form लिख लो, phosphoribosyl transferase. Okay everyone, and deficient HGPRT. Okay, deficient HGPRT is known as Leishman syndrome. Fine, this is a X-linked disease. So boys are most commonly affected. Okay, in the X-linked disease, boys are affected commonly. Girls are mostly carriers, but sometimes girls are also affected. Okay, because of some other genetic reasons, right? So X-linked disease, right? So patient presents with mental retardation. Okay, mental retardation, and because brain is most commonly affected, patient has hyperuricemia, maybe the gout, maybe the uric acid stones are formed. Now guys, why hyperuricemia? Okay, hyperuricemia because purine base are not salvaged. Okay, so produces uric acid. And one more thing is that GMP and IMP levels will decline. Okay, so there will be decrease in GMP, AMP and IMP. Salvage is absent. Okay, so leave the AMP here because we are not talking about it. So GMP and IMP, and because of that, decreased regulation of the rate limiting step. Okay, and regulation means inhibition. Okay, so this will lead to increased in the purine synthesis and uric acid. Everyone clear or not? So basically there are two reasons why this hyperuricemia occurs. One is definitely there is no salvage, so unnecessary degradation of purines every day is happening. Second, purines are made also in excess because recycling is less. So more purine, more is the degradation. Okay, because feedback inhibition is now less, very less. Okay, because of decrease in these nucleotides which are inhibitor of rate limiting step. Everyone fine? So that's, we are talking about the Lesch-Nyhan syndrome, X-linked disease, mental retardation. Okay, hyperuricemia is definitely one of the most important point to remember. Patient may present directly with the stones, with the pain in the flank region. Okay, renal colic, and next is there is a unique feature that is called as self mutilation. Okay, so patient has a behavior in which the patient bites, okay, own self, right? Nail biting, not nail biting. Yeah, nail biting is fine. Patient presents with biting own body parts. Okay, bite own body parts, and the most accessible are lips itself, okay, and the fingers. So most commonly on the fingers and lips. Okay, so they have to be kept bound to the chair. Okay, like this. Just they have to be kept bound to the chair like this, right? Because they keep on biting their fingers, they are bleeding, they are feeling the pain, but this comes like an obsession to bite. This is the feature here, self mutilation. So definitely they are feeling the pain. Okay. Right, but look at the happiness in the pain, isn't it? Okay, this is not the happiness. This patient has been edentulated, teeth has been removed. This is a severe case of Lesch-Nyhan syndrome where the teeth has to be removed because of constant biting of own body parts. Okay, Right? Not all, obviously, few has to be left, for the purpose of mastication. Okay, but front teeth have been removed. Okay. So yeah, he looks happy. Even like sometimes I feel like, when you are sitting in the class or I'm teaching, so we are also sometimes feel like bound to the chair, like this, always bound to the chair. Okay, but our hands are free. We are at least writing, but we are sitting on the chair constantly. Okay, guys, this is all about the Leschnian syndrome. Okay, so hyperuricemia, mental retardation, self-mutilation. 3 points, always remember, right? 3 points. So that's all about the purine degradation and salvage pathway. Next is pyrimidine nucleotides. Okay, so guys, pyrimidine nucleotides, they are, first of all, let's talk about the bases. So we have the 3 bases which are known as cytosine, uracil and thymine. Okay, 3 bases. कुर्सी के पैसे वसूल जाते हैं. हाँ, निसर्ग पटेल. So cytosine, uracil and thymine, right? So first of all, the basic ring structure. Okay, is a single ring. Okay, is a single ring. Fine. And there are 2 nitrogens, right? 2 nitrogens. So the numbering is like this: 1, 2, 3, 4, 5 and 6. Okay, 1, 2, 3, 4, 5, 6. Right, a number like this. So first of all, what are the sources of nitrogens? Nitrogen number 1 is same aspartate. Nitrogen number 3 is same as purine, glutamine. Okay, glutamine. And so nitrogen number 1, and everything is from like this. This is all from aspartate. So aspartate is also giving you the carbon number 4, 5 and 6. Okay, and nitrogen number 2, sorry, not nitrogen number 2, carbon number 2, that is through the CO2. Okay guys, so there is no role of glycine. Okay, if I compare it, okay, so no role of glycine. Atul Ram Prasad Yadav, yaar, please stop the horses of your imaginations, right? Thamba, please stop. Okay, so no role of the glycine is here, right? Fine guys. But if I compare it with the purine synthesis. Okay, so again let me tell you the three structures. Okay guys, this is the structure of cytosine. Okay, if you remove the NH2 you give uracil. Okay, fine, clear. And then this is added with the methyl group with the help of folate. Okay guys, so uracil, if it is added with the methyl group, then this becomes the thymidine. Okay, you got the point? So cytosine, uracil, thymidine, you know how they can be interconverted to each other. You can see that. Okay, so cytosine to uracil is deamination, uracil to thymidine is just addition of a methyl group. So you can see the structure again. Amino group was present in cytosine, uracil is oxidized, amino group is removed, and thymidine is nothing but uracil with extra methyl group. Okay, fine guys. Okay, synthetic pathway, synthesis, right? So only one step in mitochondria and remaining in cytoplasm. Fine, so let's start. It's a very simple process. So we'll start with CO2 plus the glutamine. Name of enzyme is CPS2. Okay, CPS2. Okay, so guys, what do you get is same as the urea cycle. Okay, carbamyl phosphate. Okay, carbamyl phosphate. Right, CPS2. So please write the full form. You know already, carbamyl phosphate synthetase 2. Okay, which always uses -2 ATP. Okay, one was also using -2 ATP. This is also. So guys, this is the rate limiting step. Okay, and it is inhibited by the AND product, that is UTP, and it is activated by the sugar plus phosphate. Okay, activated by the sugar plus the phosphate. Fine. Okay, so that's your CO2 plus glutamine, sugar plus phosphate. It was also activator of purine synthesis, right? Or also the pyrimidine synthesis. Now this carbamyl phosphate, okay, this carbamyl phosphate will be added with aspartate, right? So we get carbamyl aspartate. Okay, carbamyl aspartate, and the same as urea cycle, it is ATC, aspartate transcarbamylase. Okay, what do you call this? Aspartate transcarbamylase, just like OTC, ornithine transcarbamylase, aspartate transcarbamylase, right guys? Okay, अगर आपको याद है, if you remember, which urea cycle disorder had okay, I'll tell you that in in the urea cycle disorder OTC deficiency, I told you one thing. Okay, OTC deficiency. I told you in the OTC deficiency what happened, there is an increase in the carbamoyl phosphate that was in mitochondria, but that can enter here. I told you already, remember now, in the urea cycle. Okay, so this OTC deficiency was a urea cycle. This was part of the urea cycle disorder. So this carbamoyl phosphate can enter here and can catalyze the formation of, can enter into the formation of pyrimidines like uracil. That's why I told you there is a high levels of uracil or orotic acid in the urea cycle disorder, that is OTC deficiency. Okay, now guys, this carbamoyl aspartate is acted upon by the enzyme which is called as dihydroorotase, right? So this produces the dihydroorotate okay, DHO, which is now acted upon by dihydroorotate dehydrogenase. Guys, this is the only mitochondrial step, right? This is the only mitochondrial step and this is FAD dependent. FM okay, so riboflavin dependent enzyme in the mitochondria, only one step. Okay, and guys, this is the enzyme which is inhibited by a drug used in the treatment of rheumatoid arthritis. Okay, rheumatoid arthritis. Okay, leflunomide. Okay, so dihydroorotate converts to orotic acid. So guys, what I feel is this step may be important, no question till now directly, but this is the only mitochondrial step and this is riboflavin dependent. Okay, clear? मिस्टर पटेल कुर्सी का पैसा वसूल करते करते हैं. You didn't write the things, lost in between. Okay, don't mind, I'm just kidding. Okay, so orotic acid is formed, right? So guys, I told you in the urea cycle disorder there was one OTC deficiency and there was orotic acid urea, right? There was orotic acid urea, clear? So now this orotic acid, fine, this orotic acid is acted upon by the enzyme that's called as UMP synthase, which generates UMP. That's it, this finishes the pathway. Okay, so UMP is produced. Okay, so dihydroorotate dehydrogenase, right, is the only mitochondrial step. Now guys, one question which is important here, okay, and that is called as hereditary orotic aciduria. Okay, hereditary orotic aciduria. Okay, DHODH is dihydroorotate dehydrogenase. Okay, Khushbu, dihydroorotate dehydrogenase, I mentioned it, right? So also listen to me please. And the second question you're asking is OTC deficiency point is same as urea cycle point, Okay, the point was कि in the urea cycle enzyme OTC deficiency, the carbamoyl phosphate enters the cytoplasm and makes orotic acid and uracil, and that's why it leads to orotic aciduria. Fine. But now there is one more condition which is called as hereditary orotic aciduria, which is different from urea cycle disorder. Okay, how? So this is hereditary orotic aciduria, and this is due to deficient enzyme UMP synthase. Okay, deficiency of the UMP synthase. Clear? So this UMP synthase is deficient. So guys, there will be what will happen? There is decreased in UMP. So there is decrease in the other nucleotides CMP and TMP. Okay, I'll tell you how. Synthesized from UMP. Okay, so because of that there is decrease in the DNA synthesis, especially because uracil तो नहीं होता DNA में, especially because of T. Okay, so there is decrease in DNA synthesis. Now because of the decrease in DNA synthesis, what will happen? There is megaloblastic anemia, which is mild and growth retardation. Okay, which is mild. Fine. Second thing, urine has orotic acid crystals can be found, which are generally pink in color. Okay, please guys, I forgot to mention you one thing. Uric acid crystals are orange in color. Please write it. Okay, uric acid crystals are orange in color. Please write in Lesch-Nyhan syndrome. Okay, in the Lesch-Nyhan syndrome, orange powder, like you can say orange powder in the diaper, right? Orange powder in the diaper. Fine. So that you can say that is, I'll show you one image that is probably I'll be able to show you image. See this you see this, like this is a diaper. This was a case recently published somewhere, okay, in the British Medical Journal, and this is a diaper with orange powder. Okay, orange powder, and this is actually uric acid. This is not blood in the stools, this is not blood in the urine, hematuria, hematochezia, right? This is a powder form. Fine. So that's why we say this is basically a uric acid crystal. Okay, uric acid crystal. Clear? Okay guys, so that's your hereditary orotic acid urea. Everyone clear? Now what is the treatment? So guys, the treatment is oral. Okay, oral uridine, right? Oral uridine. Fine. So why oral uridine? Because we will bypass the pyrimidine synthesis. Okay, pathway. Okay, pyrimidine synthesis pathway, bypass the pyrimidine synthesis pathway. So guys, here you will not give anything else. Oral uridine will treat the megaloblastic anemia, right? Okay, you understood? This will treat the megaloblastic anemia. B12 and folic acid are normal. Okay, so you will not treat this patient with B12 or folic acid because of megaloblastic anemia if you find in the blood sample. You will treat the patient with only oral uridine. Fine. B12 and folate has no role. Right, that's it. This is all about the this point. Okay, that is hereditary orotic aciduria. Okay, so what is the next point is? Next point is convert T synthesis or the TMP synthesis. So if you take DUMP and convert this into DTMP, okay, DTMP, right? Okay, DUMP to DTMP. This I told you the difference. The only difference is just a methyl group. Okay, so we will just add a methyl group here, right? We will add the methyl group with the help of which vitamin? Methylene tetrahydrofolate. Okay, methylene tetrahydrofolate, right? Which is then converted to the tetrahydrofolate, right? I'm sorry, a dihydrofolate. Okay, so we have added 2 hydrogens also from the tetra to dihydro, na? Right guys? And the name of enzyme is called as, this enzyme is called as thymidylate synthase. Okay, thymidylate synthase. Right guys, everyone? So this is your DUMP to DTMP conversion, and we need folic acid here also. Folic acid was also required in the synthesis of purines. How? C2 and C8 carbon. Folic acid is also required for synthesis of T. T is only used in DNA synthesis. That's why for DNA synthesis there is very important role of folic acid. I'm sorry. Clear, everyone? Oral uridine will not go degradation. No, that is why it is used. Okay, that is why it is used. Oral uridine is given as a nucleoside, and it was found to be stable. It's not uracil given, it's uridine has been given, and it was found to be stable and absorbed. Okay. Fine. So thymidylate synthase is a very important enzyme. Okay, because it is inhibited by the drug which is anti-cancer 5-fluorouracil. MCQ, right? DHF should be converted back to methylene tetrahydrofolate with the help of enzyme that's called as dihydrofolate reductase. Okay, dihydrofolate reductase, and which is inhibited by methotrexate. Okay. Everyone, U to T conversion needs folic acid, right, as a methyl donor plus hydrogen donor. So donation of methyl group plus hydrogen, that's why it makes DHF. DHF converts back to methylene THF by the DHFR. DHFR inhibited by methotrexate. Thymidylate synthase is also inhibited by 5-FU. So 2 anti-cancer treatments at this point. Okay, everyone clear? So that's your that's the U to T conversion. Clear? Okay guys, fine. What are the degradation products and products? Okay, or degradation. Fine. So guys, I will not take the pathway, that's not required, it's very big, just names have been asked, you should know that. For example, from the T, thymidine, what we get is beta amino isobutyrate. Okay, beta amino isobutyrate. So I can take this as the marker of DNA turnover. Why? Because T is only present in DNA, not in RNA. I cannot take anything else as a marker of DNA turnover because A, G, C are also present in RNA. Now from the uracil and cytosine, what is the degradation product? It is beta alanine, right? Beta alanine, which has been asked as MCQ till now. Okay, so these are the two important AND products, and these are water soluble, highly water soluble, never accumulate, no disease is known, unlike uric acid, which is not very much water soluble. Okay guys, see guys, there have been a lot of MCQs have been asked in the recent times, you know, when they ask you the difference between purine and pyrimidine, okay, synthesis. So if we talk about purine and pyrimidine synthesis, I'm sorry, so the some basic differences are, first of all, you know, this begin with PRPP. Okay, so this begins with PRPP, and here the pyrimidine. Okay, I just missed that point. See guys, when we are synthesizing uridine monophosphate, come back. Okay, when we are synthesizing uridine monophosphate, so obviously you have to add sugar plus phosphate to the orotic acid. So we have added the PRPP here. Please everyone add this. So we will add PRPP. Am I clear? Right guys? So PRPP is added in the last step of pyrimidine synthesis. Right guys? So PRPP added in the last step. Okay, so one point is this. Second point is glycine is used. Here it is not used. Okay, third point is uric acid is the end product, and rest I told you. Okay, here the uric acid is not the end product, right guys? So these are the differences that you must know. Here the beta alanine is the major product, which is water soluble. Okay, so these questions have been asked number of times. Okay, recently also in the last exams. Okay, so you should know the options, right, in your mind already, like what can be the correct, what can be the wrong statement. Right guys, so that's all about the pyrimidine synthesis. Fine. Okay, so now we'll begin with DNA. Okay, so next page we'll start with DNA, then RNA, then gene expression, translation, and some techniques of DNA. Right. So guys, DNA is way beyond than just deoxyribonucleic acid. So first let's take very basic things that you already know, probably, definitely you must be knowing. So deoxyribonucleic acid. Okay, so this is the genetic material in human beings and in lots of other animals and viruses and bacteria and etc. Okay, right, only the few viruses like COVID happens to have RNA as the major genetic material, and HIV, COVID, so these RNA viruses are more dangerous, isn't it? Right. So deoxyribonucleic acid. Okay, normally what is the central dogma, as you know? Okay, genetic information travels like this. Okay, genetic information is like this, that we start with a double-stranded DNA and we get a single-stranded RNA, right, by the transcription, and then we get the protein. Fine, and we get the protein. So this central dogma can be bypassed, okay, by the viruses. So only the viruses can turn RNA to the DNA using some enzymes like reverse transcriptase. Okay, fine. So RNA can be converted to DNA. So viruses do not follow the central dogma of the genetics. Okay, viruses, some viruses, not all, like those which are RNA viruses, they can make DNA. Right guys, so this exception. So this was a recent MCQ, but you know already, at least right now you remember, right? Probably by the time you cover up your MBBS, you remember this or not. So that is the revision part. Okay, so exception, viruses and RNA is converted to DNA. Okay guys, now DNA. Silly doubt, what is difference between thymidine monophosphate? Yeah, thymidine is a nucleoside, I can say, like basically. I said thymine, remember thymine, which is T, and TMP is the thymidine monophosphate. Okay, means nucleoside plus single phosphate. So thymidine is a nucleoside. Okay, just like cytidine, just like uridine. Okay, just like adenine. Fine. Okay guys, let's take some basics of DNA. Guys, DNA was discovered by Watson and the Crick and Wilkins. Okay, Watson, Crick and Wilkins. Okay, in the year of 1953, they used a technique that is called as X-ray diffraction, okay, or which is also known by the name X-ray crystallography. graphy which is still used to identify the structures. Like suppose you want to know the structure of the COVID-19, you will use this technique. Okay, so the virus can be visualized. So this is a technique which is a highly precise technique to see the molecular structures. Okay, there are a lot of other techniques now available, but it is still used very commonly. So what is the name of technique? X-ray crystallography. Okay, so actually they got Nobel Prize, and actually this technique was actually DNA was visualized by Rosalind Franklin. She visualized the DNA, but she died before the Nobel Prize was announced, and Nobel Prize are never given to the people who are dead. So there's no name of her in the DNA. I mean to say, we always know Watson trick, Watson trick. That's it. Okay, just for knowledge. Okay, so DNA is double stranded, as you know, right? And double stranded, the two strands are anti-parallel, as you know, right? I'll tell you the exact meaning, but first I'm just writing one strand is 5 prime to 3 prime, and other strand is 3 prime to 5 prime. And I told you it's only the sugars which are in different direction. Okay, so anti-parallel. Okay, and some more basic points. Okay, let's understand this anti-parallel meaning and the bonds in DNA. Fine. So guys, as you know, this is the DNA. DNA is a polynucleotide structure. Okay. Gregor John Mendel, no, he was not related with DNA at all. He studied chromosomal genetics in the pea plant. Okay. Okay. So DNA is a polynucleotide structure. Fine. So what is the meaning of that polynucleotide? Let's see. So this is the base present here, suppose A. This is the phosphate present here. So mononucleotides are there. Okay, polynucleotides. But we use what in the DNA? DNA is made of DAMP, DGMP, DTMP, and DCMP, deoxynucleotides. So deoxy means only H, no OH. Okay, on the second prime position. Okay, so the incoming nucleotide is added like this. Okay, so this is the phosphate. Okay, let's say this is G. Okay, this is a G. Just to understand, this is H and this is OH. Okay. Okay. So H and OH. Clear? Then this phosphate and OH, okay, they will bind together, okay, by the phosphodiester bond. Please write the name, phosphodiester bond. Guys, this is the function of DNA polymerase, right? Okay, this is the function of DNA polymerase. Okay, the phosphate is bound to the OH. Clear? So this is one of the very important bond here, and see, this is starting from 5' phosphate. Okay, this is ending in the 3' OH. So what is the direction of this strand of DNA? 5' to 3'. Fine. Now similarly the other strand will be in opposite direction. Okay, so if we make the opposite strand, okay, like this, suppose this is the T here on the first prime base, and this is H, this is OH. A bind with the T with the 2 hydrogen bonds, right? And similarly this is also, okay, this is C, and these are the 3 hydrogen bonds. This is H, this is OH, right? Okay, this is the phosphate and this is the phosphate. Okay, fine. So this OH and this phosphate, they are linked like this, and this is again the phosphodiester bond. Okay, this is the phosphodiester bond. Clear to all? Right, guys? And these are known as the hydrogen bonds. Hydrogen bonds, as you know already. So this is the direction of the DNA. We are starting from 3' OH and ending in the 5' phosphate. So this is how the two strands run in opposite direction. Okay, the two strands are running in the opposite direction. Clear? Okay. Frederick Sanger. Right, guys? Okay, the bond between here, I'm just writing what is the charge here is nothing but this is called as base stacking force. Please write it like I'm writing here, BSF. Okay, so what is BSF? So the AT and GC base pairs have the base stacking forces. Okay, base stacking forces, which are nothing but hydrophobic force plus van der Waals force. So guys, remember all the forces which I have mentioned here. These are part of MCQs. Okay. Right, guys? And what is the charge on the DNA? As you all know, this is negatively charged due to phosphates. Okay, right? DNA is empipathic. Okay, please write empipathic means both hydrophilic and hydrophobic. Okay, hydrophilic and hydrophobic, empipathic. Okay, then the DNA which is discovered by Watson and Crick, it is having a right-handed helix. Obviously the double helix. Okay, it's a double helix. So if we talk about the basics of double helix, okay, then just some more points, basics, okay, like this. So guys, this is the DNA, suppose, and this is the groove that you can call minor groove, which is having lesser depth. Okay, one is called as major groove. This is the major groove. This is the diameter of the helix. Okay, diameter of the helix, that is around 20 angstrom or the 2 nanometer diameter of helix. Please write the word diameter of the helix. Okay, minor groove, major groove, right? This is one turn, 34 angstrom, and having the how many base pairs? 10 base pairs per turn. Okay, so A, T, G, C, the base pairs. So total base pairs are 10. Okay, there are total 10 base pairs per turn. Clear to all? Okay, so that's your the basics of the DNA, basics of DNA structure. Fine. Now there is a special type of DNA that is called as Z type of DNA. Okay, Z type of DNA, Z for zigzag. Okay, Z type of DNA is something which has 12 base pair per turn. That's more compact DNA it is. Okay, and it is seen there is no major grooves, so it is arranged in such a way that it has no major grooves. Okay, it is rich in G and C, right? And it is seen during transcription. Okay, when the RNA polymerase, okay, seen during transcription when the RNA polymerase separate the double-stranded DNA. Clear to all? Which words are difficult here? Okay, so there are no major grooves, rich in GC, seen during the transcription when the RNA polymerase separates the double-stranded DNA. Okay, got the point? So that is a Z type of DNA, right? Kani Revant. Here I am talking in English only. Which words are difficult to understand? Let me know, please. Okay. Clear, guys? So this is not the normal DNA. Normal DNA that we see that we have discussed. Okay, so that is called as what type of DNA? That is some bDNA. Okay, so normal cells have the B type of DNA as discussed above. Okay, so which is the DNA that we have in our, it is compact, so the major grooves are removed. Okay, so it is packed like in such a way that only minor grooves are visible. Okay, so normal cells have the B type of DNA as that we have discussed above. Fine guys. Okay, so now the next point is, so guys, DNA is very big. Now we'll see the DNA packaging, that how the DNA which is very big is packed in a small nucleus, okay, with the help of histone proteins. Okay, with the help of the histone proteins. Right. So now our next topic is DNA packaging. Please write the heading DNA packaging. So DNA, the total 23 pairs of chromosomes that we have. Okay, as you all know, 23 pairs of chromosomes. Right-handed helix and left-handed helix, Astha, what do you mean is like if you look at the, if you have seen the direction of a helix in a screws have helix. Okay, so the one which is moving towards the right hand, the direction of the helix, that is moving towards the right hand. Actually we can see only the 3D structure, not here. Okay, you never got it, so you now try to find a screw at your home. Okay, and look at the direction of the something which is going away from you. Take the screw like this, okay, pointed and towards you, right? And if it is going away from you, that is called as right-handed helix. Usko bolte hain right-handed. And if you think it is coming towards you, right, the helix direction, that is left-handed helix. Actually no need of that, I mean not required, but if you want to really understand. So 23 pairs of chromosomes, this makes 3 billion base pairs, okay, with average of 2 meter length. Okay, average length is 2 meter. Okay, so guys, the DNA is very big. Okay, and it is, I told you already, negatively charged. So how it can be packed? So now we have proteins which are known as histones. Okay, now because such high amount of DNA and nucleus is very small, right? Nucleus is, let's say, 6 microns in the diameter, 6 microns in the diameter. Okay, so we need something to pack it compactly, which attracts the DNA and packed it. So histones are positively charged proteins, right? These are positively charged proteins. Okay, Why they are positively charged? Because they are rich in two basic amino acids, arginine and lysine. Okay, so they are rich in arginine plus the lysine. Okay, so that makes it positive. And what are the types of histones which are present in organisms, including humans? H1, H2A, H2B, H3 and H4. Right, so these are the types of, okay, H1, H2A, H2B, H3 and H4. So these are the types of histones that we have total. Okay, so now what we will make first, we will make the octamers. Okay, octamers. Okay, histone octamers. Right, so how to make a histone octamer? So we will take 8 histones together except the H1, right? So we take 2 molecules each of H2A plus H2B plus H3 plus H4 multiplied by 2, right guys? So this is, so there is no H1. What was the MCQ? There is no H1 in octamer, right? So guys, now we'll make okay, now we will make the nucleosome. Fine, so there is something that is called as nucleosome. Nucleosome is histone plus DNA, that is called as nucleosome, and this is the fundamental unit, basic unit, you can write like this, basic unit or fundamental unit of DNA packaging. Okay, fundamental unit of DNA packaging, that's called as nucleosome. Let's see what is that. Okay, so it is like this. So suppose this is the octamer. Fine, this is black color that we make is octamer, and let's say this is our DNA in red color which is wrapped over like this. So this is DNA. Okay, 146 base pair of the DNA. Okay, right, so DNA 146 base pair of DNA wrapped around the histone octamer in 1.75 turns. Highlight everything, 1.75 turns. How much base pair? 146 base pairs of DNA. Guys, this is what you called as core nucleosome or the nucleosome, but H1 is still missing. So where to use H1? Actually this is the core nucleosome, right? So we will make one more structure that is called as 10 nanometer fiber, okay, or beads on the string appearance. Okay, beads on the string appearance. Fine, what is that? So this is one octamer, this is a second octamer, this is a third octamer, right? Like this. Fine. This is DNA which is wrapped, DNA which is wrapped, DNA which is, let's say, wrapped like this. Okay, and here comes the H1 histone. Okay, this is the H1 histone. This is the H1 histone which is present on the linker DNA. Okay, linker DNA. And guys, this is the diameter, that is 10 nanometers. Why we call it 10 nanometer fiber? Because 10 nanometer is the diameter, okay, of this octamer plus. Okay, you got the point? Linker DNA. Fine. See, what I am telling you is we are packaging DNA. So how to pack the DNA? Okay, first of all, you understand this. Just a minute. I'll tell you. Look at this figure from Harper. Fine. So guys, how the DNA is present in our cells nucleus? It is present as chromosome. So I am telling you how the chromosomes are actually made. Fine. So to make this chromosome, you had the naked DNA first of all. On this naked DNA, you combine the octamers. Okay, octamers, which makes a 10 nanometer fiber. Right, this makes a 10 nanometer fiber, and then we will keep on packing it. So negatively charged DNA, positively charged histone, they bind with each other very tightly, right? This makes the DNA nucleosome. This makes the structure known as nucleosome. Okay, is that clear now? Okay, everyone clear now? So actually, if you take the smallest portion of the chromosome and you visualize this in the electron microscope, okay, when the DNA is separated, right, not compact. Compact means it will be visible as metaphase chromosome. When the DNA is not in metaphase, right, and it is loose, okay, then it can be visualized as the beads on a string appearance. Okay, what is that? Octamer plus the DNA. Is it clear now? Okay, so this is what we are doing. So you should remember that how it is made. Where is the H1? You can see there is H1. Okay, you can see H1 is present on the linker DNA. This is the linker DNA. So this structure is called as beads on the string appearance. Everyone clear? Okay, so it is definitely the negatively charged DNA and positively charged histone proteins which are binding together, making the 10 nanometer fiber. And this is in the electron microscope. You will find this as the beads on the string appearance. Okay. Fine. Beads on the, right guys? So this is how the this will keep on DNA will be compacted finally as chromosome, right? So we get ultimately later on 30 nanometer fiber. Okay, next level of packaging, right? Then we get some loops, then we get rosettes. Okay, loops, rosette, okay, and finally chromosomes, which are visible in the metaphase stage. Okay, understood? So this is how you understood, this is how the chromosomes are made. Clear, guys? Okay, so that's how the DNA is packaged. Okay, DNA is packaged like this, and DNA which is tightly packed, okay, is known as heterochromatin. Okay, and DNA which is loosely packed is called as euchromatin, right? So you already know two terms. One is called as heterochromatin. Okay, so there is a tightly bound histone and DNA, right? Histone, H plus means histone, please write histone plus DNA. And then second is, this is inactive DNA, and second is euchromatin, right? This is loosely bound, okay, loosely bound histone and DNA, because of charges that I will take in epigenetics section again. Okay, epigenetics section, I will take this point again. Okay, one more thing that I want to tell you is that okay, so basic is this heterochromatin and euchromatin. Okay, fine, guys. Here I am taking a lunch break. Okay, I am taking lunch break right now, right? So come back in 35 minutes. Is this fine to you? Come back in 35 minutes. I'm taking the lunch break because I am feeling hypoglycemia. Okay, so see you in 35 minutes, right? Then we'll cover up till 3. Okay, see you. Okay, so moving to the next point. So guys, we'll talk about first of all the basic topic, important topic, and that is the DNA replication. No, tomorrow is not last, 9th is last. Okay, fine. DNA replication. So we'll take DNA replication in prokaryotes and eukaryotes. Okay, so DNA replication. So guys, DNA so first of all, in prokaryotes and then in eukaryotes we can take, both are important. Okay, till now the most of the questions have been asked on prokaryotic replication because that was a basically first one to discover, right? So let's take it. So we'll start with, okay, so suppose guys we have a double stranded DNA. Okay, so this is the double stranded DNA. Now guys, prokaryotes have circular DNA. Suppose bacteria has a circular DNA, but just to understand, we will take it as a because in the eukaryotes I will only mention the names of factors. which may be asked in future exams or which has been asked some of the exams, right? Has been already asked, right? So double stranded DNA, 3 prime 5 prime 5 prime 3 prime. So those nucleotides which are A and T, they are weak compared to G and C, right? So they separate first. So first of all we use the enzyme that's called as helicase. Okay, helicase. So helicase is ATP dependent enzyme, right? It is a ATP dependent enzyme. Okay, so this helicase will separate the DNA, fine, generally at the AT point, right, A and T point. So this will start separating the DNA, like, let's say, like this. Okay, stop the discussion please and listen to the okay. So this is your double stranded DNA, okay, 3 prime to 5 prime, 5 prime to 3 prime. Okay, so this is the separation has started. Okay, now during separation, okay, so DNA supercoils, right? DNA supercoils, and for removal of these supercoils, what is required? For this required the enzyme that's called as topoisomerase. Okay, so guys, topoisomerase relieves the super coiling of the DNA. Okay, super coiling of the DNA. Okay. There are two types of topoisomerase actually. One is topoisomerase 1 and topoisomerase 2. Okay, topoisomerase 1 and the topoisomerase 2. See guys, 1 and 2, what is the basic differences? Topoisomerase type 1, see, basically the DNA is supercoiled. Okay, DNA is supercoiled like this, let's say this wire. Okay, let's say this wire that you can look at this. This is the DNA. Suppose it has been highly compacted and this is supercoiled. Fine. So type 1 topoisomerase breaks only one strand. Fine. Okay. So the ligases, they have DNA topoisomerase, they have two functions: cut plus seal. Okay, cut and the seal, right? So see, this is the cutting, nick means cut. So after cutting, this has been straightened like this for the replication to move forward, right? And then it has been joined. Okay, then it has been joined, clear, like this. Okay, so we have joined it, understood? So first there was a cut here, okay, then it has been joined. So this is type 1. So single strand break, type 1, and if I do it on two strands, it's type 2. Function is same, removal of supercoiling, and function is same again, ligase plus nuclease plus ligase, cut and seal, nuclease plus ligase. Okay, so function stays the same, right guys? So single strand breaks, double strand breaks, fine. Okay, and topoisomerase, there are some other differences that we can write now. Okay, so basically this is topoisomerase 1, topoisomerase 2. Okay, so topoisomerase 1 is, so it has nuclease and ligase action. This is same. Okay, so single strand DNA breaks and reseal. This is double strand DNA breaks and reseal. Okay, then this is no ATP used. Remember, because it is breaking both the strands, we can say ATP used, doing more hard work, need some energy, right guys? Okay, right, so no ATPs are used, and here again ATPs are used. This is present only in eukaryotes, right? This is present in both, okay, in both the eukaryotes and prokaryotes. Okay, both the eukaryotes and prokaryotes have this type of topoisomerase. Clear? Topoisomerase is present here. So in the prokaryotes, prokaryotic type 2 topoisomerase is also known as DNA gyrase. Okay, it is also called as the DNA gyrase. Fine, which is inhibited by the ciprofloxacin group of fluoroquinolones group of antibiotics, right? Okay. Okay, DNA gyrase has a special property that it can induce the negative supercoiling, okay, and apart from removes the positive supercoil. Now again, positive supercoil means when the DNA is twisted in the same direction as it was previously. Okay, so that is what you call as a positive supercoiling. Okay, fine, sorry. So it can induce the negative supercoiling and positive induce the negative supercoiling. That is something unique of this, because topoisomerase is supposed not to induce any supercoiling. It is supposed to remove it, but it can induce it. Okay, how this is important for bacterial survival is not known, but it is somehow important in the growth of bacteria in the next round of replication. That's not clear, right? How? Okay guys. So what are the drugs which inhibits topoisomerase? They are called as etoposides. Okay, etoposides, right? Okay, these are the name of drugs, right guys? Okay, so now the next is, what is the next step? So now we have the double-stranded DNA. Okay, like this. Suppose upper one is 3 to 5, 5 to 3. So guys, first of all, helicase, then topoisomerase. What is the next one enzyme? So next enzyme that we take is known as primase. What is the other name of primase? It is called as DNA dependent RNA polymerase. Okay, so what is the function of the primase? It make a short RNA primer, makes a short RNA primer. Primer means to initiate. Okay, what is prime? The priming is to initiate. Fine. Okay, so like you are right now you are primed, okay, for studies, okay, because you don't know what will be the type of questions they may ask, but whatever they ask here, the questions will be, the topic subject will be the same, the questions pattern may change, that's it. Okay, so never think that what will be the next pattern of exam and everything, it doesn't matter, right? Because things will be same. They cannot change the name of DNA polymerase, or they cannot change the the structure of cornea or the ECG, right? Okay, waves in ECG, so everything will be same. You just have to read, continue reading, and think nothing about it. Okay. Fine. So next what we are doing is we are simply adding a primer. Okay, so primase, DNA dependent RNA polymerase make a short RNA primer to initiate the replication, as DNA polymerase cannot initiate on its own. Am I clear? So this is the reason. Fine. So how the primer acts? Okay, so let's see. This is our DNA which is in the process of separation. Fine, like this. This is 3' to 5' strand. Suppose this is the enzyme which is called as primase. Okay, it binds here. Primase enzyme binds here. Okay, let's say this is the primase enzyme which bind here, and this primase enzyme makes a 5' to 3'. Okay, 5' to 3'. Fine. Done. I already scrolled up. Prokaryotes with single stranded DNA. I really don't know which prokaryote has that. I'm really sorry. If anything is like that, then single stranded DNA will not will be again needing a topoisomerase 1 at least. Okay, if it is there. Fine. You getting a point? So update me on that please later on. So 5' to 3'. So guys, what is the direction of any polymerase? Any polymerase, you don't know the name of that polymerase. What you need to remember is they are always in one direction, and that is 5' to 3' direction. Okay, so the previous old strand, parent strand, is 3 to 5, so you will start from 5 to 3 direction, right? You will make a short primer. Okay, primer. Then this is taken care by the DNA polymerase 3. Okay, so this is the DNA polymerase 3 which got the 3' OH group. Okay, so this 3' OH, okay, because we are making ultimately you are adding nucleotides, so the last nucleotides 3' OH will be now free. Okay, so basically the point is the DNA polymerase 3 add the nucleotides to the free 3' OH group. Okay, free 3' OH group. I mean to say here, ultimately whatever the number of nucleotides there may be, but ultimately this OH group will be free at 3' position, okay, of the primer. Suppose this was the primer. Okay, right, any base is there, so you will be this bacteria will be able to add one more nucleotide here with the help of DNA polymerase. Is this clear? Okay, so free 3' OH of the OH group. So now the DNA polymerase 3 will start making the DNA. okay, from this 3' OH group, because it is not separated beyond this point, so I'm stopping here. Okay, DNA is still not separated, right? There are still bonding. Okay, helicase is breaking the hydrogen bonds, so it is not reaching to the still in the way of breaking. So helicase use ATP, it separates the DNA. Okay, it basically break the hydrogen bond. I'll tell you a lot of disorders names which are because of helicase deficiency in eukaryotes, human beings. Okay, like Bloom syndrome, I'll tell you, for example. Okay, so we are talking first prokaryotes. Fine. So primase, DNA dependent RNA polymerase makes a short RNA primer, I told you. So it is not separated beyond this. So this is my currently 3' end. Okay, currently 3' end. Now guys, this strand is already in 5' to 3' direction. Okay, and we start the synthesis of DNA from 3' end of the parent, as you can see. तभी तो 5 to 3 होगा, like then and then only 5 to 3 DNA you can make if you have the free parental 3' end parent DNA, okay, or the old DNA strand. This is the old DNA strand, okay, but its 3' end is not free, it is not separated, na. So then, you know, I will take this as the available 3' end. Okay, so let's see. Okay, this is called as leading strand. That is, what do you call this? This is called as the leading strand. So the new strand which has been made is called as leading strand. Clear? Now lagging strand. Okay, so lagging strand, okay, or the Okazaki fragment synthesis. Okay, lagging strand lagging strand is synthesized on the 3' to 5' parent, I'm sorry, on the 5' to 3' parent strand. Okay, 3' end is not available for primer synthesis. So take the open 3', okay, open, very important word, open 3' end for the primer synthesis. Okay, so we will take the open 3' end for the primer synthesis. Clear, guys? Okay. Right, guys, so that's your open 3' end for primer synthesis. We'll take that. Okay, so we'll make small fragments now. So suppose this is the DNA which has been separated only this much. Okay, this I will take, though this is already 5' to 3', right? This is the open 3' end. Okay, primase binds here. Okay, so suppose this was a primase, bind here, makes a small primer, then DNA polymerase 3, again bind here, DNA polymerase 3, and this makes a 5' to 3' strand. Clear, guys? 5 prime to 3 prime strand. Everyone clear? Okay, so we made only short strand, right? Okay, and we'll keep on making such short strands once the whole DNA is separated. Fine, suppose the whole DNA has been separated. This was upper strand 3 prime to 5 prime. Okay, so everything has been separated. So we will have multiple primers, okay, and multiple short strands. Okay, guys, and everything will have a direction, and that is 5 prime to 3 prime, 5 prime to 3 prime. Clear? This is the way. So guys, these each strands are known as short or Okazaki fragments. Everyone clear now? And the upper strand was already like having a primer, and if it has been separated completely, then it will be the leading strand, continuous. Okay, leading strand is continuous. Open 3 prime end. Okay, somebody is asking me, open 3 prime end is this, na? Till now DNA is not separated, so I will take this part as the, ये तो 5 prime हो गया, this is now 3 prime end, I will take it as. Okay, so I will start making DNA from this direction. Okay, so that's called as open 3 prime end. This is the actual 3 prime end of the DNA strand, which is not available. So we'll start from here. That's what we did. We made a primer and a DNA. Again open, little bit more open, it's opening, and we keep on making short DNA fragments. Okay, so can you see both the strands are made by DNA polymerase 3. Okay, now some important points. First of all, the DNA polymerase 3 has 5 prime to 3 prime polymerase activity. Correct? All the polymerase have same. It synthesizes both leading and lagging strand. Very important points. End of the day you should remember these points. Okay, lagging strand. Okay, DNA polymerase 3 also has 3 prime to 5 prime exonuclease proof reading action. Okay, 3 prime to 5 prime proofreading action. Okay, so has 3 prime to 5 prime exonuclease proofreading action. Okay, what is the meaning of the proofreading action? Okay, understand this. Proofread means to remove the errors. Okay, what is the meaning of proofread? Proofread means like to remove the error, editors. Okay, edit, that is editing is known as proofreading. Okay, so what are the mistakes are there, you have to remove them. That's called as proofreading. Okay guys, so how to proofread basically is 3 prime to 5 prime. See, word exo means from outside and nucleus. Okay, so let's understand this. See here, so this was the, okay, let's make it again. So let's say this was our the strand of the DNA, okay, which was in 3 prime to 5 prime direction, and what we started making the leading strand. Okay, we started making the leading strand. So first we made the primer, because without primer DNA polymerase cannot act. I told you, DNA polymerase needs the free 3' OH group of the primer to initiate replication. That's why it is called as primer. So primer provides the 3' OH group. What is the function of making primer? It provides a 3' OH group. So when we are making the DNA, so we are putting like this: A, then T, G, then C, C, G, like that. Suppose in the end, here, just to understand, in front of A you put C, right? This DNA polymerase 3 has done a mistake and has put C here. So that is a mistake. It has to be removed. So guys, which direction we are moving on? 5' to, this is our 3' direction. Okay. So what is happening here? We are removing now we have to remove C. C is not required now. I want to change it. So basically, suppose this is the DNA polymerase, what you can say, 3. Okay, so DNA polymerase 3 will stop here. This is the DNA polymerase 3. Now guys, this DNA polymerase 3 will move in the reverse direction. Okay, so move in 3' to 5' direction and remove C from the 3' end. And means exo, remove means nuclease. Okay, so suppose this is a DNA strand. In my hand you can see, and this is the end where the wrong base is there. So DNA polymerase has also a action of exonuclease. So it will go back and remove the C. So 3' to 5' direction, and cutting from the end is exo. Okay, cutting from the end is exo. If I cut from somewhere, endo, right? If I cut always from ends, exo. Got it, everyone? So this is called as 3 to 5 exonuclease proofreading action. Okay, so it removes and again replace the correct base, right, which will be the T. Okay, everyone clear? Very simple. Okay, so what is the next point? See guys, directions are important, names are important. Okay, next is role of DNA polymerase 1. Okay, so removes, it removes the RNA primer via, what is, I have written DNA polymerase 3. This is exo, means end. Okay, so next, remove the RNA primer via the 5' to 3' exonuclease action. Okay, 5' to 3' exonuclease action. So guys, see this, very simple. Okay, see, I want to remove this primer, green color. Okay, so I want to remove this green color primer. So I have to grow for I have to use this enzyme from outside. So exonuclease for which direction? 5 2 3. Done like this. Okay, so 5 2 3. We are going in direction 5 2 3. Remove all the primer. 5 2 3. Okay guys, everyone, because RNA है यार, we don't want RNA in the DNA, so we have to remove the primer now. There is no need of primer now, no? It is RNA actually, ribonucleic acid, right? So that's why we have to remove primer, and this is the direction. Very, very important. Okay, this is absent in DNA polymerase 3. Very important point again. Okay, absent in DNA polymerase 3. Fine. And then DNA pol 1 replace the primer with DNA via 5' to 3' polymerase action. Okay guys, okay, via 5' to 3' polymerase action, and plus also proofread via 3' to 5' exonuclease action. So guys, 1 and 3 differ only in one direction, and that is primer removal direction. Clear? Understood? So DNA polymerase 1 has this extra activity which is not found in DNA polymerase 3. Rest of the actions are same, right? Okay, one more point is, which is has been asked number of times in all India names, and that is there is a term that's called as clinofragment. Okay, so what is clinofragment? Clinofragment is DNA polymerase 1 without 5' to 3' exonuclease action. Okay. Yes, all the points of polymerase 1 I am writing. Okay, we are discussing polymerase 1 now. So clinofragment is the DNA polymerase 1, fine, without 5' to 3' polymerase exonuclease action. But they are like, when to use such a fragment? You know, this is artificially created. Okay, this was artificially created for the PCR reaction where the primer is not removed. It is not used nowadays. Okay, right? Got it, guys? It is not used nowadays because now there are better enzymes which even can remove the primers. It is an old discovery. It is a man-made enzyme where one of the fragment of the DNA polymerase 1 was removed by the mutations created in the genes, and this was helping in PCR reaction. Okay, so like that. So you can skip this point, you only remember this point. Okay, For you, this is the point, that without polymerase 1, without this. Okay, so that's your DNA polymerase 1. What is next point? So next is ligase. Okay, joins the Okazaki fragments, okay, via obviously the phosphodiester bond. So yeah, like if we see what is the function of these polymerases, ligases and everything, primases, obviously to make polymer by which bond? Phosphodiester bond. Okay, what is the difference between clinofragment and DNA polymerase 3? Now there is a difference, a lot of differences are there. One of the difference that you can understand is DNA polymerase 3 is a highly processive enzyme, very fast enzyme, and which acts on both leading and lagging strand for synthesis of DNA routinely. Polymerase 1 is very slow enzyme and it acts only to remove primer and replace it that with the DNA. Very small sections are replaced with it, and that too slowly. Okay, so it's not a processive enzyme, it's very very slow enzyme. Okay, first you write this now. Okay, so it's not same, it doesn't mean that you have removed it and it becomes same. No, they're still having different affinity, different speeds of action. Ligase joins, though a good question. Okay, very good question. Okay, so ligase joins the Okazaki fragment via phosphodiester bond. Okay, obviously on the lagging strand. So guys, ligase or the DNA ligase only acts on lagging strand, not on the leading strand. Okay, not on the, because yeah, there was only one primer and that was removed, that's it. Okay guys, so that's your DNA ligase. Okay, now DNA polymerase is highly processive, that I told you. Okay, okay, I can mention that also. Fine. DNA polymerase 3 is highly processive enzymes. Okay, now what is processive? 3 more than 1, definitely. Okay, what is the meaning of this term? Because recently one of the students asked me the meaning of this processivity. Processivity means the number of nucleotides added, okay, to the DNA without dissociating, okay, from the template strand. Okay, so they are like this, they actually like this, so they are bound and they keep on moving, so they dissociate in between also, they keep on moving, dissociate sometime. So how many number of nucleotides are added without being dissociated from the template strand? Okay, understood? So that's called as processivity in the bacteria or in the eukaryotes. So moving on to the next page. Done everyone? You guys are done, everyone? So I am moving to the next page so that it doesn't hang. Highly processivity. Okay. Now guys, in the eukaryotes, eukaryotic replication in short, that you should know. Okay, so first of all we start with again the double stranded DNA. Okay, it is so big that it has multiple points of origin. Ori, that's what you call it, multiple points of origin. Fine. So we'll draw a flow chart and then we'll understand this. Simple, same process. Multiple points of origin are there because it's very big. Fine. Then first of all, definitely there will be some helicases. Okay. So helicase enzyme will be there. Helicase enzyme always use the ATP. Helicase in eukaryotes are named like this: WRN, BLM, BLM. I'll tell you, these are names of some disorders: Werner syndrome, Bloom syndrome. Okay, so just write, these are also the recent MCQs. Multiple points of origin. Helicase is the name of enzyme. Then we also need the topoisomerase 1 and 2. So that I told you already differences. Then the primase. Okay, so not primase actually. First of all, then DNA polymerase alpha, okay, binds at the 3' of the both, I told you. Okay, so polymerase alpha has 2 subunits. Okay, and has primase plus DNA polymerase action. So guys, there is no separate primase in in the eukaryotes. It is part of something called as DNA polymerase alpha. Okay, so DNA polymerase alpha has primase activity and also the polymerase activity. It is a multiple subunit, 2 subunits. Okay, then primer is made. Okay, then next is guys, there is a protein that's called as RPA. Okay, RPA prevent the re-annealing or fusion of the 2 DNA strands. Because your base pairs are same, na? A can again bind with the T. How to prevent them? So there is some proteins which are unique here in eukaryotes like RPA. Okay, so this is the one point. Fine, and then you should know what is the next point is. Then next is polymerase switching. Alpha is not able to work further, right? It is very slow enzyme. So next is polymerase switch. okay, where the Pol alpha okay is replaced by Pol delta. Okay, Pol delta on the leading strand. Please change it. Pol epsilon on the leading strand and Pol delta on the lagging strand. So guys, these are the two important points, that eukaryotic leading strand is synthesized by DNA polymerase epsilon and delta is synthesized by delta is making the lagging strand. Are you clear? What WRN and BLM are the names of helicases, and I'll explain it later, just wait. Okay, I told you these are names of helicases. Okay. So next is polymerase switch is done. Alpha is being replaced. So there was 3 on both the strands, DNA polymerase 3, DNA polymerase 3. Not here. We have epsilon on the leading strand, delta on the lagging strand. Okay. There is a protein known as RFC. RFC binds the DNA strands and loads pCNA. Okay, pCNA. See guys, full forms of these all are not required because they will create a lot of confusion. Okay, fine. So just remember just short names, and these are not direct questions till now. Only questions that I'll tell you are the names of the eukaryotic polymerases. But if we look at the whole process, it will be easier for us. Okay. So RFC binds the DNA. There is a protein known as RFC. Okay, as a factor RFC you can write, binds the DNA strands and loads the pCNA. It's an antigen actually. Okay, loads pCNA, and guys, this pCNA is a clamp. So you know what is clamp, right? Clamp. Okay, so pCNA is a clamp which make the polymerase delta epsilon processive. Okay guys, I told you now what is the meaning of processive. Processive means when the when the polymerase is moving. Okay, polymerase is moving without any dissociation. Okay, it just keep on adding nucleotides without any dissociation from the DNA. So in eukaryotes, in us, this pCNA, okay, this pCNA makes the DNA polymerase processive with high speed, low errors. Okay guys, understood? So remember at least pCNA name. Forget RFC, it's okay. RFC is just loading it. Okay, so first alpha, then comes the Pol delta or epsilon. It's simple, right? Leading epsilon, delta, lagging is delta. Okay guys, and next is, okay, so DNA ligase, again that joins the Okazaki. Before that, just a minute. First of all, you have to remove the primer now. Okay, so primer removal is different here. RNAs and FEN1, the full form of which is flap endonuclease. So can you see the name? It's endonuclease, not the exonuclease. because guys, exo ends of chromosomes, okay, in the eukaryotes will be because there will be two, but origin of replications are multiple. So we don't say a removal from end, it is removal from in between. Right? Are you getting a point? Okay, so flap endonuclease, we use the term. RNase H, RNase H, and flap endonuclease removes the primers, and then finally DNA ligase fuse the Okazaki fragments. Okay, they fuse the Okazaki fragments. So guys, that's the eukaryotic polymerases. Okay, so if I talk about again the eukaryotic polymerases, so guys, likho alpha, I told you alpha makes the primer. Okay, there is one beta that we have not discussed directly. It is used in DNA repair. Okay, most importantly base excision. Okay, base excision DNA repair. Okay, alpha and beta, they have no proof reading activity. Recent PGI Chandigarh exam MCQ also. Okay, alpha and beta has no proofreading. Proofreading is 3' to 5' exonuclease action. Okay, so alpha and beta, they have no proofreading action. Clear? Then there is gamma. Gamma is exonuclease, please, exon nahi, exonuclease action. Gamma. See, if I write gamma, okay, so there are multiple M's. M for mitochondrial DNA synthesis. Okay, just M for mitochondrial DNA replication, gamma. Okay, then delta, I told you, lagging strand. Okay, and epsilon is lagging strand. Here to all guys. So these are the eukaryotic DNA polymerases. Delta being the most processive enzyme, epsilon and delta. Okay, and which makes the eukaryotic DNA polymerase processive? PCNA. Okay, PCNA. Antibodies in against PCNA can form in some disorders, like in SLE, systemic lupus erythematosus. What happens? There are antibodies formed against the human nucleus, human DNA. Antibodies are formed. Okay, anti-nuclear antibodies, anti-histone antibodies, antibodies against PCNA. That you will read in your path or medicine lecture. Okay, so that's the eukaryotic part of the. But guys, there is a problem in the replication in eukaryotes. And what is that problem? Right? I'm sorry, I wrote it wrong. Thank you. Okay, epsilon is, that's what I mentioned previously. Okay, yes, epsilon is reading. right? Now next is telomere shortening, which is part of replication only. Summarize, okay, summarize. See, in prokaryotes, we have two most important enzymes, DNA polymerase 1 and DNA polymerase 3. Okay, DNA polymerase 3 is the major enzyme of replication in both leading and lagging strands. Okay, DNA polymerase 1 has activity of primer removal, and that is 5' to 3' exonuclease. Okay, 5 to 3 exonuclease. Fine. Then primer removal is always 5 to 3. Polymerase is also 5 to 3. Right? But proofreading is in opposite direction, 3 to 5. Now what is proofreading? I am writing something and again I am going back and checking here ki I have to publish it. Kuch galat to nahi hai. That is proofreading. So go back and check. Polymerize kiya maine. I have written an article and I went back and check everything else. So that is proofreading. So that is 3 to 5. Okay. Ligase always joins the Okazaki, and that's 2 only on the lagging strand. Okay, so like that. So that is in prokaryotes. In the eukaryotes, we start with helicase and topoisomerase, and then alpha, which has primase activity, which is then taken up by later on by the, क्योंकि थोड़ा बहुत polymerase activity is also present in alpha. So alpha also makes a small DNA strand, but then delta take over on the lagging and epsilon takeovers on the leading strand. And RNase H and flap endonuclease removes the primer in eukaryotes. Okay, so PCNA was recently asked. Okay, and see, mitochondrial, out of all these, some questions have been repeated number of times, including recent exams. For example, this very important is required in DNA repair. This is important, required for mitochondrial DNA synthesis. Human beings have two types of DNA, nuclear DNA and the mitochondrial DNA. Okay, understood? Okay, telomere. What is telomere? These are the ends of chromosome. Okay, with okay. So these are the ends of chromosomes. Okay, and they have a typical sequence as shown in this figure, that is A triple G double T. Okay, so with the A triple G double T repeats. Okay, ends are protected from the nuclease mediated attacks, okay, by a protein that's called as Shelton protein. Okay, Shelton protein. So guys, देखो ऐसा है कि it is like that DNA should never be free. Okay, you got my point? DNA ends should never be free. Okay, so if DNA ends are free, they will be attacked by the nuclease enzyme. That's why till now the gene therapies are not very much successful, because if you introduce gene from outside, it is a free DNA, it will be degraded by the nuclease enzymes. Okay, got the point? So that is your telomere first of all. So telomere ends are protected by some proteins, okay, which are known as sheltrin. And ends are also, like, if it's a double-stranded DNA, ends are also actually folded like this, okay, like this. And there are certain proteins which are present at the end, which are known as sheltrin complexes, sheltrin protein, okay. So guys, the point is that telomere shortening, okay, during replication. So telomere, or the ends of the DNA, right, ends of the DNA shortens, okay. Ends of the DNA shortens with each replication cycle, okay. And this is the tick tock, the clock, the mitotic clock of the cell, the mitotic clock of the human body, or the death, okay. Okay guys, fine, you got my point. So telomere are the ends of chromosomes, and they shortens with each replication cycle. Right, each replication cycle. Now let's see, first of all, let's understand why it is shortening, okay. So guys, this is as the last primer is not replaced with the DNA due to, just write it first, okay, unavailability, okay, unavailability of, okay. So the last primer, please don't disturb me guys, okay. ये कौन लोग है प्लीज, मारिया, अब्बासी, है ना, मानसी, प्लीज you can guys you can leave if you're done, right, please leave, okay. You go to the rasoda and eat something and sleep. ठीक है. So, and those who are listening, please keep on listening, okay. I'll explain this, that why this telomere shortening occurs and what is the importance of telomerase enzyme. Okay, so as the last primer is not replaced with the DNA due to unavailability of the free 3' OH group without DNA polymerase cannot work, okay. DNA polymerase cannot work. You got the point? Now see guys, let's see this, I'll explain this, the replication part. Okay, so first of all, this was the 5' to 3' strand, okay, 5' to 3' strand, fine. We already made primers like this, I told you, okay. Then we had some DNAs, understand this, and otherwise it will not be clear, okay, that how this occurs, fine. So we start with, so this was our 5 to 3, and so this is again 5' to 3', and this is again 5' to 3', and now guys, if I remove the primer, okay, so now there is primer removal, So after the primer removal, what we are left with? This is again 5 prime to 3 prime. Draw it. Okay, 5 prime to 3 prime. All the primers removed. Fine. So now we are left with this DNA strand, and this DNA strand. So these are again in the same direction. 5 prime to 3 prime. 5 prime to 3 prime. 5 prime to 3 prime. Is it clear, everyone? Okay, these are all Okazaki fragments that are supposed to be joined by the ligase, but before that you have to replace this primer. Okay, replace this primer. So guys, all the primers can be replaced. Suppose this space can be replaced because 3 prime end is available. Okay, so using this 3 prime end of the DNA, suppose this space is filled. Using 3 prime end of the DNA, this space is filled. Okay, you're getting the point. But this space, because there is no 3 prime end available here, you cannot fill this space. Okay, there is nothing is available. This space is totally empty now. So how will you fill this? Okay, you cannot fill this space, right? Okay, or just understand that, you know, because of unavailable 3 prime OH group, DNA polymerase cannot work. Okay, so 3 prime OH is not available and it is not working, right? So this will lead to a structure which is, let's see, like this, simple, like this. So this is 3 prime to 5 prime, 5 prime to 3 prime. So guys, this is 3 prime over hang. Okay, 3 prime over hang. Fine. And this is called as telomere shortening, right? And this lead to aging and death. Okay, this lead to aging. So guys, why we age? Because with each happy birthday we are losing some amount of telomere or the DNA. Okay, so why we age then? What is the problem? The problem is we are losing DNA. So if we are losing DNA, we are losing genes for normal function, like for the functioning of kidney, liver, bones, muscle, etc. Okay, so that's why our body is aging. Okay, so telomere is shortening, ends are keep getting shortening slowly and slowly and slowly. Okay, you got the point, guys? Fine. So that's your shortening of the telomere. So that's why we age, guys, you know. So telomere can be maintained if there is enzyme that's called as telomerase, and that's a very important MCQ. Guys, I told you Hayflick limit already. You know, we cannot survive, our cells cannot survive more than 50 to 70 replication. That is Hayflick limit of the cell. Human body as such has billions of cells, so can survive, right? So telomerase is an enzyme, right, which prevent the end shortening. Okay, which prevents the end shortening. First we'll write, then I'll explain this with the figure. Okay, so which prevents the end shortening. Okay, and this is the problem is it is absent in all the somatic cells. This is the big point, that we don't have telomerase. Okay, we don't have the telomerase that is in all of our cells. Okay, it is absent in all the somatic cells. So in our body we have two types of cells, and like actually we can say three type of cells. One is a somatic cell, one is the stem cells. Actually stem cells have this telomerase activity. Okay, so stem cells have telomerase, so that's why they don't grow old, stem cells. Okay, and we grow old with each happy birthday, and that's what we celebrate, no? So stem cells have telomerase. Which stem cell? For example, bone marrow stem cell, right? We have stem cells in the skin. Okay, skin. Okay, then germinal cells, germinal epithelium. Okay, so which give rise to ultimately sperm and ovum. Okay, so they keep on dividing, but they don't age, right? Like whatever is the age of mother and father, or the male and female, the child's age will be zero only, right? Because the sperm has donated, or the ovum has donated the DNA without shortening, without loss of any gene, without loss of any genetic material. Okay, right? So aging is there in the somatic cells, but not in the stem cells. Okay, bone marrows are producing the same stem cells throughout the life, unless until there is a disease. Okay, so like that. So that's your absent in all somatic cell and present here. Okay, now guys, what is the telomerase enzyme is actually, and I'll tell you how it acts, which is not important, but I'll explain it so that you understand. Telomerase is RNA plus proteins. Okay, right? It is also known as RNA dependent DNA polymerase. Okay, RNA dependent DNA polymerase. Fine. Okay. Yes, who is this? Skin. Yes, skin has stem cells. How it is replacing epidermis every time? Okay, it is replacing the epidermis even after your minimum injury or the maximum injury. The skin is being replaced. How? Right. So that is how. No? Who is this? S for skin. Okay, so RNA dependent DNA polymerase, right? Or it is also known as, it is also called as a reverse transcriptase, right guys? Reverse transcriptase, very important name, very important name. Okay, let's see how it acts. Okay, and one name of disorder. Okay guys, let's see how it is acting. Just a minute. Okay, see guys, this is the end of the chromosome. Okay, this is the end of the chromosomes, right? These are ends of chromosomes which are shorter. So see, this is a repeat. Okay, 3' overhang, 3' overhang, right? This is the telomerase enzyme, blue color. It binds here. It has a RNA, which you can see this 3' to 5' RNA. So taking this RNA as a template, okay, taking this RNA as a template, what happens? This a nucleotide sequence has been synthesized here. right? Okay, so a nucleotide synthesis has been synthesized here. So guys, actually what happens is, you know, the telomerase has extended, right? The telomerase enzyme has extended a new nucleotide sequence on the already long DNA strand. See again, it was already long DNA strand, and with the help of the RNA as a template of telomerase enzyme, we have again make, you know, strand of the 3' and longer, so it becomes more long, right? So actually telomerase is not synthesizing directly the shorter strand of DNA but already long strand of DNA, and now this is the primer, okay, which has been made by the DNA polymerase alpha in front of the elongated strand, and rest is simple, right? So this will make the DNA. As you can see, DNA polymerase will keep on making this whole strand of DNA, and end of the day we will remove the primer which was made here. Okay, so primer which was made here will be removed. So this is how at least we get what was short. Okay, so and in the end what we get is the shorter strand has been elongated. Clear guys? So you just try this. Okay, understanding this again, fine. Because you have the next class, so I'm finishing here. Fine guys, so see you tomorrow, and I will repeat this process again, telomerase, right? Okay, so thank you so much.